Good afternoon, everyone. Welcome to the MELS graduate student thesis presentation talks. I'm Mike Charette. I have the great pleasure of being the MELS coordinator. So this is kind of where we get a lot of our incoming graduate student class to really shine as the superstars that they are. The overall philosophy of today is for them to present what they propose to do as their thesis project. So the majority have started in September, so therefore they're really just kind of getting on with getting familiar with the lab of the thesis project. So to keep the day moving, because we have quite a long schedule, I'm going to just launch into it and introduce the first speaker. So the first speaker today is Carson MccDonald, who graduated from BU in the spring with a major in chemistry. I remember teaching Carson quite a bit. He then joined the MELS graduate program. I think we were all very excited about that when he did that under the supervision of Dr. Kieran Taylor in the Department of Chemistry also. So Carson's talk today is entitled Clean Hydrogen Fuel Production via RH catalysis. So take it away, Carson.
Thank you very much, Dr. Charette, and thank you everyone for being here. So as mentioned, what this presentation, what my project kind of is trying to do is finding a way to make hydrogen in a clean way using rhodium-based catalysts. So currently, fossil fuels such as gasoline, diesel, etc., are required for pretty much everything that we do. A couple examples. First, generating electricity. This is actually a prime example here in Brandon at the Brandon Generating Station, just out by the dump in the east end of the city. The Brandon Generating Station has two natural gas turbines. Those turbines use natural gas, methane, as a fuel that is burned in order to generate electricity. Now that the temperature is getting colder, everyone's been heating their homes now. Lots of homes here in Brandon use gas furnaces, which again take natural gas, burn it, and in turn produce heat for our homes. Supply chains are also heavily dependent on fossil fuels. The ships, the boats, the planes, the cars that get our food from the field to the manufacturing plant to the store and back to our homes for our meals all depend on fossil fuels. So just using these few examples, we can see that fossil fuels are highly, highly important to both us as individuals and as economies as a whole. But there are some problems with fossil fuels that we do face. Fossil fuels are a non-renewable energy source. It takes millions and millions of years for the Earth to naturally create fossil fuels. Because there is technically a finite amount of fossil fuels in the Earth, every time we take fossil fuels from our planet, the reserve that we have gets depleted. But the big issue with fossil fuels is that they are a key contributor to climate change. For example here, combustion, which is the burning of a hydrocarbon or a fossil fuel, produces greenhouse gases. An example reaction here uses methane, natural gas, or CH4. When burned in the presence of oxygen, produces moles of water as a product, as well as our carbon dioxide. Can you still advance? Yep. Carbon dioxide, seen there in red. Because when we burn hydrocarbons, it produces CO2, which is a greenhouse gas. This enhances the greenhouse effect on our planet, which is when radiation from the Earth's surface goes back into the atmosphere and then gets re-radiated back down to the surface. If there's more hydrocarbons being burned, there's more CO2 being produced. Therefore, the greenhouse effect is enhanced, causing temperatures to therefore rise more. So is there a viable solution that we have or are going to have to the fossil fuels that we are so heavily dependent on right now? Well, there may just be. Hydrogen. Hydrogen fuel is seen as one of the primary candidates for the replacement of fossil fuels. Hydrogen, as you can see right here, the first element on our periodic table in the top left-hand corner. The reason why hydrogen is seen as a good candidate is because it has a very high energy density. It's renewable when the hydrogen is produced in a sustainable and eco-friendly way. It's combustible, just like our fossil fuels. We can take our hydrogen fuel and combust it just like those fossil fuels to produce energy for us. It's highly, highly available. Over 75% of the universe by mass is actually made up of hydrogen. And the big, big plus of hydrogen is that it is clean. When we burn hydrogen, the only byproduct that we have is water. As I showed on the previous slide, when we burn a hydrocarbon, we get that water, but we also have a greenhouse gas such as carbon dioxide as a byproduct as well. So when we burn our hydrogen, water is our only product. But there are some issues with hydrogen. Currently, the way that we produce and store and use hydrogen is actually kind of dangerous. Hydrogen is required to be compressed at very high pressures in order for us to store it effectively. These high pressures enhance the risk of things like explosion, and others have other safety concerns. Also, hydrogen production is currently not very clean, actually. The majority of hydrogen that we do produce currently is made through a process kind of simplified right here. We take our good friend natural gas, our fossil fuel, when heated in excess of 800 degrees Celsius and in pressures of excess of 20 bars, and reacted with water, we produce our hydrogen there on the right, which is what we want for our fuel. But as a byproduct, we also have carbon monoxide, again, another greenhouse gas. This is referred to as gray hydrogen because though the hydrogen that we do produce is going to be used cleanly, the way that we produce it, because the carbon monoxide is a byproduct in this process, we're not really getting ahead. We're still having a greenhouse gas as a byproduct of this process. So is there a more green way that we can produce hydrogen compared to what we have the way that we produce it right now? Well, there may just be. Clean hydrogen production via transition metals is something that has been thoroughly investigated as a clean alternative to hydrogen production, or a clean way to produce hydrogen. Transition metals, which are highlighted in yellow here, which are the D block, if you look at our periodic table over there. Transition metals have been proven capable catalysts for both hydrogenation and dehydrogenation. Hydrogenation is the storage of hydrogen, where dehydrogenation is the production of hydrogen fuel from some sort of hydrogen source. In this simplified reaction right here, we have our metal highlighted in purple. When reacted with some moles of water, we produce a cationic metal, or a metal with a positive charge, as well as hydroxide ions, and as we can see, our hydrogen fuel there on the right. As you can see, compared to the natural gas way of producing hydrogen, there are no greenhouse gases produced via hydrogen production via transition metals. There's none of that carbon monoxide or CO2 produced. This is why it is referred to as green hydrogen, because there are no fossil fuel byproducts on the product side, on the right-hand side of our reaction equation. Also, with transition metal catalysts, we can store hydrogen fuel at atmospheric pressures. One of the concerns that I mentioned before was that right now, hydrogen fuel needs to be condensed at high pressures, which gives an explosion risk. If we were able to store our hydrogen fuel at atmospheric pressures, we kind of eliminate that safety component, that safety risk. So a lot of focus on hydrogen fuel production via transition metal catalysis has been around our group 9 metals. Our group 9 metals, there again in the D block, consist of metals such as cobalt, rhodium, and iridium. The main metals that have been looked at or focused on have been rhodium and iridium for catalysis for hydrogenation and dehydrogenation. When these metals are paired with a ligand, where a ligand is something that attaches to a metal center, it kind of supports the metal center, provides electronic support to either the rhodium or the iridium. When our group 9 metal is when the catalytic complex is has a ligand included, one ligand of focus has been 1,2,3,4,5 pentamethyl cyclopentadiene, but nobody calls it that because it's way too long. It's just CP star. So our CP star here has been a primary ligand of focus for hydrogenation and dehydrogenation. When it's bonded to the metal center, it's referred to as CP star. Before we bond it to the metal center, it's referred to as CP star H. As you can see here, this is our CP star. It's a 5-membered ring with our 5 carbons making up that ring. And on each of the carbons of the 5-membered ring, there's a methyl group or a CH3 group that is bonded to it. CP star is a tripod ligand, meaning that it bonds kind of 3 times to the metal center. It bonds through these pi electrons right here in the double bonds, as well as this lone pair of electrons right there. And that's how it bonds 3 times to the metal center. Here's a quick example of a CP star group 9 metal complex that maybe could be used in hydrogen catalysis. On the top there, we have our CP star fragment. Though it's not shown that it actually bonds 3 times, that's what this kind of circle represents right there. The circle represents that the tripodicity of the ligand. Here we have our metal center, shown in purple. Here's the rhodium-3 metal center. And then down at the bottom, we have, these are called pyrazole ligands. Those 2 5-member rings with the 2 hydrogens. Those are pyrazoles. They're bonded as kind of spectator ligands. Spectator ligands are included in these complexes to provide some sort of catalytic support, either through bond breaking, bond forming, etc., or providing electronic support to the metal center. Also, as you can see at the top right-hand corner, this is a cationic complex because of that positive charge. And in order to create a coordination vacancy, 1 chloride had to be removed. So rhodium-3 is kind of happy with 6 substrates coordinated to it. We have 3 from our CP star. We have 1 from the chloride and 2 from the pyrazoles. That's 6. The precursor that we get the rhodium from has 2 chlorides on it, meaning to create one of those coordination vacancies that is required for bonding to the metal center, we have to have one of the chlorides removed. So as kind of a theme of something that I've learned in my short time doing research is that you think you got something, but then a big issue comes up and it's probably not right. So originally, just like everything else, CP star has to have an issue with it, obviously. CP star originally was thought to be innocent. What this means is that because it was a tripod ligand, it provided a lot of electron density towards the metal center, making the metal center more electron rich. Because of this tripodicity and the 3 bonds that it has to the metal center, it was originally thought that CP star was chemically inert. What I mean by this is that they thought originally that it didn't provide any kind of, it didn't have any role in bond breaking, bond forming. It was just kind of there to support the complex, provide electrons to that metal center. But later on, CP star was proven to be more non-innocent. What I mean by that is that CP star was actually found to be chemically involved in hydride delivery. Though this was a very interesting finding, what also was found was that the non-innocence of CP star can actually lead to catalytic degradation. Because the CP star is involved in hydride delivery, CP star is able to be hydrogenated, meaning that hydrogens will bond to the carbons of that 5-member ring, and which could cause the pi electrons and the lone pair of electrons to be kind of redistributed, meaning that the CP star fragment may actually no longer bond to that metal center. So some of our current work that we're working on right now is based off of kind of 2 different papers. There was someone named Miller in 2016. They were one of the original founders of CP star non-innocence. Miller was one of the people that kind of put forward the mechanism for CP star non-innocence. What Miller said was that CP star was the preferred site actually of hydride delivery, not the metal center. But my supervisor, Dr. Powell, then showed just last year via density functional theory, which are computer-based calculations that are ran for reactions. He found that CP star through these DFT calculations was actually not the preferred site. It was the metal center. So currently what I am working on is the experimental validations to Dr. Powell's findings. What we're going to try to do is experimentally validate whether CP star is the preferred site of hydride delivery or if the metal center is, and try to push the reaction in either direction. So first off, I synthesized a few CP star rhodium complexes for investigation in this study. I've successfully synthesized a more electron donating complex because of these methyl groups. Because of these methyl groups here, this makes the complex more electron donating, pushing electron density closer more towards the metal center. I synthesized on the other end an electron withdrawing complex. BPDA was attached. These sulfonate groups will pull more electron density away from the metal center. And also for comparison purposes, I synthesized successfully a more neutral complex just for comparison purposes. Also, just recently, the other day, we found out we have successfully synthesized a complex using one of our own synthesized ligands. So the complexes on the previous slide all had ligands attached that were commercially available, so they're pretty much all pure already. The synthesized ligands, I actually synthesized this ligand with the help of Dr. Powell, bis-pyrazole methane, bis meaning 2 pyrazoles on that methane, attached to that methane center. And then successfully, we attached this ligand to the metal center. And we're going to do the same as the same thing we're going to do with the complexes on the previous slide. We'll check their viability for hydrogenation and dehydrogenation for hydride delivery at the CP star or at the metal center. Next stage we're currently on is analysis of complexes. We primarily use proton NMR down on the second floor for our kind of first analysis of things that we made. We've also been very grateful to receive a new liquid chromatography mass spectrometer from the Chen lab on this floor, which we are very grateful for as well for some LCMS analysis. Once we have analyzed our complexes, we're going to attempt catalysis with them. So here, catalysis is kind of broken down in this diagram right here. Here we have our complex with our with our either chloride or bromide. In this case, it will be a chloride, filling that coordination vacancy at the metal center. It will then move into that chloride will then be removed using a silver complex that will create a coordination vacancy at the metal center. There in the middle, you can see our metal hydride complex. And then what we're going to see as well is if that hydride delivery occurs at CP star from the metal center. So in the end, in this kind of mini project, we're going to determine which is preferred. If the metal hydride complex is preferred for hydride delivery, or if CP star is the preferred site of hydride delivery. Later, in more of the grand scheme of this project in Schrin's NSERC proposal, we're going to be looking at alternatives to CP star complexes. So CP star, as I mentioned before, can become protonated both under basic conditions and at high pressures. This can cause catalytic degradation, as seen here in this picture. So as our CP star fragment slowly gets protonated, we lose these pi electrons to that protonation. Therefore, CP star may not actually bond to the metal center of our complex. So catalysts that we may produce that exclude CP star may actually have longer lifespans and more turnover because they will not become protonated, and they'll become less likely to kind of break down. So some of our later work we're going to work to synthesize rhodium-based catalyst that excludes CP star from the catalytic complex. And what we're going to do with that is attempt something called bidirectional catalysis. What I mean by that is that catalysts that can both produce hydrogen as well as store hydrogen. So hydrogen production using these catalysts will be via liquid organic hydrogen carriers, such as methanol and isopropanol. When methanol here at the top and isopropanol at the bottom of the picture are dehydrogenated, they form formaldehyde and acetone respectively as their dehydrogenated substrates. On the other hand of the bidirectionality of these catalysts is hydrogen storage. And with these complexes, we'll be able to hopefully store hydrogen at atmospheric conditions, again, eliminating that safety component that is an issue right now with hydrogen fuel. And again, because we won't have a CP star fragment, we're looking at possibly more catalytic turnover and a longer lifespan for these catalysts. And quickly, just to touch on our research process, we like to call it computationally directed synthesis. What this means is that first, Shrin usually runs density functional theory calculations of reactions on his computer. If DFT tells us that a reaction may work, we go ahead and into the lab and synthesize it. If we successfully synthesize our complex, we move on to the analysis stage. There's that LCMS that we've just been gifted from the Chen lab, as well as on the right, we have our nuclear magnetic resonance or NMR machine that's down on the second floor that we use for primary analysis. Quickly, my acknowledgements to my supervisor, Dr. Pal. I don't think he likes the picture. To Busu for funding my trip to the Canadian Society of Chemistry conference back in June, and as well to NSERC for Shrin's discovery grant that he and I are both very grateful for. And with that, I'll take any questions.
So I'm just up here to moderate the question period, so I'm going to encourage you to address your questions directly to the student presenting them, and I'm just going to keep an eye on the time and stand up here.
How common is rhodium? Sorry, did I just do a question? Very rare. Yeah.
Not very common, but more common than things like platinum.
Okay, so like from a scaling up perspective, is this going to cause, like, is this scalable globally, like this type of technology thing?
I would say,
Like, I'm not certain how much, what kind of masses you're using or would be used to do this.
most of the reactions we do right now, just to check the viability of the reaction, we're using usually about 100 milligrams
Okay.
of a rhodium precursor. So in a way, yes, it is scalable up, but depending on expenses, I'm not an economist, but it'd be up to them to decide.
Right.
Okay.
Okay, I gotta look at the out table elements sorted out. So on the one slide it said that rhodium and radium have been like commonly used, I guess. Is there a reason that cobalt hasn't been used? I noticed it was also on the slide. I'm not sure if you mentioned that.
Just the.
You guys friends?
I think it has been investigated. I'm not really 100% sure. And every metal kind of has its thing that it does. Just the reactivity. I think cobalt is much more stable than those other two. There could be a reason why it maybe isn't as good as the other thing.
Assuming that was not a setup question.
It was not.
No. Usually 3D, but the highest row in the transition block, those metals are really hard to control in terms of the reactivity. So cobalt can leave hydrogen, but then it goes blockers and it starts hydrogenating every single fragment in your so your ligand will get hydrogenated, your substrate will get over hydrogenated. It's really hard to control.
Okay, just to keep us on time, I think we're going to stop the question period here for Carson. However, if you've got additional questions, maybe you can chat with him during the coffee period or afterwards. So thank you very much. So, the next presenter today is, Anna Cushon. She grew up on an organic farm in southeastern Saskatchewan, where she spent 3 summers working for a PhD student, from the University of Manitoba. And I can only imagine that the interaction with that PhD student is maybe what led you here.
A little bit, yeah.
Okay, I figured. So the PhD student that she was working with was looking at phosphorus uptake in the wheat. So she then went on to complete her Bachelor of Science in Biology at Providence College in Manitoba in the spring of 2024, and is now focusing on peatland and hillslope hydrology under the guidance of Dr. Pete Whittington in the Department of Geography and Environment. She's also one of these superstar people who's a Bobcat on the women's basketball team. So her talk today is entitled Into the Moonscape, Sudbury, Ontario's Regreened Forests and Their Impact on Hillslope Hydrology and Peatlands. Take it away.
Thank you. Today, I'm going to be taking you to the moon. No, not the real moon, but somewhere that is closer to home, but once was equally as barren. And that place, ladies and gentlemen, is Sudbury, Ontario. And I know what you're thinking. Why should a Manitoban like yourself care anything about those Easterners out there? And I'm with you. But I promise you, there is relevance to this research, not only for people in Ontario, but for all Canadians and citizens of this earth. So what even is a peatland? It is a wetland that accumulates peat. Peat being soil that is made up of dead plant material that is built up slowly year after year. And because of the waterlogged nature of peatlands, it does not decompose fully because of the anoxic conditions that are created. So you can get this meters upon meters of dead plant material. And sphagnum moss is a bit of the star of the show in peatlands and is largely responsible for many of the unique natures of peatlands. And so you can see there in the upper pictures just what sphagnum moss looks like. And so peatlands are important, like other wetlands, because they have the ability to purify water, mitigate floods and droughts, and also support great numbers of biodiversity. But what sets peatlands apart is their ability to store carbon. And they do this, and they're able to store 30% of the world's terrestrial carbon is stored in peatlands, despite making up only 3% of the Earth's surface. And they do this as all those plants have taken up carbon dioxide through photosynthesis, as plants do. But since the waterlogged nature, they don't decompose fully, or they decompose very slowly over time. So you can get potentially thousands of years of carbon built up. And so, unfortunately, it is a double-edged sword with this storage of carbon, because you can have these great stores, but it can also be released if peatlands are disturbed or drained or harvested. And unfortunately, that's what's happening in Sudbury, Ontario. They have been disturbed. And so Sudbury is located in northern Ontario and has historically been one of the worst sulfur dioxide and metal particulate pollution emitters. And years of pollution led to severe effects on the plant communities, which earned it its names of the Moonscape. And you can see here, a lot of the area was clear cut for mining, but then because of the sulfur, the soils and lakes were all acidified, which had very harmful effects on all the plant communities. And so it was so bad that in the 70s, they got a visit from astronauts on the Apollo 16 mission, training to go to the moon. And no, they weren't just pretending they were on the moon in Sudbury. They were actually looking at shatter cones and geological deposits left behind when the asteroid hit Sudbury all those many years ago. But it didn't help Sudbury's reputation as a moonscape. And yes, they did look that cool. And so eventually people began to take action, and there was a call to action. So began the Sudbury Regreening program in the 1970s. And it involved universities, industry, government, and everyday citizens working together to regreen Sudbury. And they did this using a basic recipe. So first, to neutralize soil acidity, they added lime before fertilizing the soil to bring back all those nutrients needed for growth, before they planted a seeding of ground cover, before finally, a year later, trees could be planted. And Sudbury has had great success in their regreening, and 30,000 hectares have been reforested since the 1970s, and that's about half that still needs to be done. So I joke that Sudbury is still a moonscape, but it really has made tremendous bounds to regreen the area. And you can see here just a comparison of the landscape in 1980s to 2008, just the tremendous difference. However, peatlands are still yet to be restored. And Sudbury is home to 6,000 hectares of peatlands, and they're the current most dense and potential carbon store in the landscape. And however, because of the contamination from the metals, they're not acting as carbon stores anymore, and they're actually releasing in some peatlands up around 100 years of sequestered carbon. However, in some of the most contaminated peatlands, this amount is approximated to be up to 2,000 years. However, if all the peatlands in Sudbury were healthy and functioning as they should, they could store up the same amount of carbon in 2 years that all the regreened forests have in the last 50 years. And that's not to say that the forests aren't important for the ecosystems, but it just shows you just the amount of carbon that these peatlands can store. So the forest did also have a positive impact in other ways on the lakes in Sudbury. So Sudbury is also a city of lakes. There's about 330 lakes greater than 10 hectares within this greater Sudbury area. And because of the liming that took place on the uplands, this helped to neutralize the acidity of the lakes. So because of runoff helped carry lime during the early days, it really positively impacted the lakes, as well as the planting of trees helped to further prevent erosion, which helped to improve water quality as well. So for the lakes, the healthy uplands resulted in healthy lakes. But that isn't quite what we're seeing with peatlands. And so here we get a good overview of the landscape in Sudbury, and here's our peatland. Unfortunately, not unfortunately, but peatlands are naturally acidic. So that is part of what makes them unique. But what's good for the soils and the lakes, which is lime, is not so good for the peatlands. And we can see, and we are starting to see, differences in vegetation along the edges of the peatlands here. We see different species, more like marsh-like vegetation that we would expect to see there. And so we want to examine how this runoff is affecting the peatlands. And we also, our major goal on this project is to restore the peatlands back to their full carbon sequestration potential. But to do this, we need to really understand the water balance in these peatlands and how that is affecting the restoration. As we discussed, by their waterlogged nature, that is really how they store the carbon. And so we need to understand, to fully understand the water balance, we need to understand the amount of runoff that is getting to these peatlands and how the regreening of the hill slopes is affecting this runoff. So that is our main question. And we want to examine how has the evolution of hill slopes through regreening affected runoff to the peatlands. And we want to look at how the natural restoration, so no treatment being applied, has evolved versus the active restoration. So as we were looking through and trying to determine these study sites and these hills we're going to have to look at, we wanted sites that were comparable in slope and aspect, in proximity to a smelter, so they have the same relative contamination levels, as well as proximity to a peatland, and looking at the year lime and planted so we can compare the evolution. So we went to the Sudbury Regreening database, which shows you, if you can go to this site, you can actually click and see when each of these locations were limed and planted. So I also included just a little bit of a smelter. So those are the 3 main smelters in Sudbury area. And so looking at this map, it took me like just poring over this map and trying to think maybe like the Coniston area would work for our study. But luckily we had another reliable database in the form of Peter Beckett, who is one of the many great faces behind the Sudbury Regreening program and has an amazing ability to recall just the landscape and different sites we could use. And so he recommended in about 5 seconds that Coniston would work for this study. And he also recommended Daisy Lake Uplands as a potential site. He also let us know that in the 80s, there was actually a study done on the effects of surface liming of soils on streamflow chemistry. So there's actually hydrological data that has been done in the 80s that we can now compare from at the same, we can go to the same hillslope and compare how runoff has changed since the 80s. So this was big news for us. So going out to this actual Coniston Creek site, it's really easy to see the differences in the restored landscape versus the unrestored. So on the right, we see the conifers, which were the main type of trees planted in the regreening, and they're nice and green, obviously. But on the left, we see birch trees, which have all lost their leaves as this was taken in the fall. But birch trees are one of those pioneer species that can colonize areas and are a bit more tolerant to those to that acidity and disturbance. So at the Coniston Creek area, the lower level of the basin that was examined was limed, but the upper level was never limed. So when we look again just at this comparison here, this is a bit of a rough drawing of how the basins will look, but we're going to be comparing the upper unlimed basin to the limed lower, as well as this is another side, so another catchment that has a restored upper and a restored lower. So going down to the ground level of what these hillslopes actually look like, you can see some big differences just in the obvious vegetation levels as well as soil levels. And so that gives you a clear picture of and start to imagine what the runoff amounts would look like at each of these slopes. So now going to Daisy Lake, another one of the sites we would like to look at. It was restored in the 90s, so 30 years ago, and it has 2 main catchments that have been studied. And only one small area has been limed, and the rest has just been left to naturally restore, and has yet to be restored. So again, we can just see an overview of this area. This is just the small section that has been limed and restored. And then also as we were walking, we could see exactly where they had made their line of where they wanted to apply lime on one side, and then the other side was just left to restore. So that was kind of an interesting find. So again, just a rough drawing of what we will be comparing, but we have the unlimed upper and unlimed lower, as well as restored upper and unlimed lower. Now again, just these ground level hill slopes, we see very similar hill slopes. So now I want you to think about this again in a little more cartoony way, which is very easy to picture how runoff will change at each of these sites. So I want you to imagine runoff on a hill slope like this versus this versus this. As you can imagine, there's going to be a lot more runoff on the first site, which is what we kind of imagine and what we see as those naturally restored sites, versus here where there's lots more soil as well as plants to capture and slow down this runoff. So runoff is controlled by a variety of factors. I didn't actually include all of them, but these are the main kind of things we'll be focusing on. So rainfall intensity, evapotranspiration, soil type, and saturation level. So if the soil is, or the rock is impermeable, slope and vegetation cover. So those are the things we are going to be looking at and measuring. So how will we do this? Well, first we need to measure precipitation amounts, as precipitation is the main driver behind runoff. And so here we will use tipping bucket rain gauges, and this is a weather station that was actually at Daisy Lake, and that's our little tipping bucket rain gauge that looks a little worse for wear, but that will be how we measure that. We also want to look at how much water is getting intercepted by trees as it falls. So we'll have two sites for the tipping bucket rain gauge, one out in the open and then one placed under a tree. Then we want to look at how much water is not going to be contributing to runoff, so that some of the water will be leaving the system through evaporation. And we can measure evaporation using lysimeters, which are basically just a bucket of representative soil and vegetation, which we can weigh to determine how much water has evaporated. So looking at how much water is actually entering the soil, we can use a soil moisture probe. Some of the water will also not enter runoff into and will instead be caught in small depressions like this one. One of the ways we can measure this is just by pumping out these depressions and measuring the volume. One of the ways we can measure through flow is using an eaves trough to a bucket, and that will give us a rating of discharge. Another way we can measure discharge is using a V-notched weir. So when we're walking out, we actually again stumbled upon one of the original weirs that was used in the 1980s. It's also looking a little bit worse for wear here. That's what an actual V-notched weir will look like. But essentially they measure discharge, so it's a volume through time. And what we will want to compare is not discharge, but runoff, which is an aerially weighted measurement of discharge, so we can compare it along the different basins at their different sizes. So one of the ways we can analyze runoff is through a hydrograph, which just shows the change in runoff over a period of time after a rain event. So when we look at A, we can imagine one of those naturally regenerated slopes, so no treatment was applied. We can imagine it will be what is described as flashy. It's going to run off very quickly and be done very quickly. Whereas we think of B as a regreened slope with lots of vegetation, where that water is going to get stuck in various places and take its sweet time getting down that slope. Another way we can compare runoff is just by comparing the evolution through time. So our first points will look at the runoff that occurred in the 80s versus today. So first looking at the, taking it back to the Coniston Creek site, we'll first look at the unlined upper level. So that site will naturally have a higher amount of runoff just because of its position on the slope. And we expect to see a little bit of a decrease just because of an increase in vegetation that will naturally occur. Next, for the restored upper level, we would expect to see a sharper decrease in runoff due to that greater amount of vegetation. Then to the line lower, we expect to see already, again, a lower amount of runoff to begin with, just because of its position on the slope. And then a sharper decrease because of the effects of liming on the vegetation. And then finally, the restored lower is what we see to have, or what we expect to see to have the greatest decrease in runoff, just because of its position, as well as the greater amount of vegetation that we expect to see. And thus, so these results will give us a picture of the runoff and how it is arriving and affecting and how much of it is getting to the peatlands. Oh, and these are just some examples of numbers we just estimated approximately, and again, just showing the greatest decreases in runoff. So in summary, peatlands are great carbon sinks, and we need to protect them. In Sudbury, unfortunately, there are current carbon emitters because of contamination. However, However, water table is the most main control on biogeochemical processes in peatlands. So in order to restore them, we need to understand the hydrological properties of these peatlands and the hill slopes. So this includes understanding the effects of regreened hill slopes evolution on runoff arriving to the peatlands. And so our hope is that peatlands in Sudbury can be used as an example for restoration, not only in Sudbury, but around the world, and can help our fight against climate change, bringing a new hope for peatlands. Thank you.
Okay, wonderful talk. Questions?
So is wetter better for carbon sequestration?
Yes.
So we should just deforest?
Some might argue that, but no. Definitely, obviously, the trees do have an important impact for ecosystems, but
Let's say we don't care about the other, we just care about the land. But they're really what's important, right?
yeah, pretty much. But yeah, like they need to have a certain water level in order for the decomposition not to occur. So if they start to dry out, they do start to decompose faster. So it is important that they are wet. Yes.
But too wet, we get more methane.
Right. You want just the right amount.
So what do you characterize sunlight or temperature during these trends? I mean, I'm sure there's some general differences over the years in terms of how that's affecting, especially evaporation or runoff or things like that.
Yeah, we will definitely be taking temperature measurements as well as just annual like precipitation, comparing that as well for sure.
Yeah, great presentation.
Thank you.
I'm a biologist, right? So I'm just curious, do we know anything about how much of the diversity being lost when that happened? And with this goal out, like end goal is to revert back the vegetation, impact on climate change. Are we getting back the diversity there, like the biodiversity? Because they're responsible for carbon emission and management of the ecosystem.
Yeah, great question. Our hope is that yes, like the biodiversity will come back as restoration occurs. I'm not too.
You could say I don't know.
Yeah, that's not my level of expertise yet, but I don't know if you know.
There has been tons of work on all aspects of the regreening. The peatlands really are one of the last main parts of the system, so. Okay.
Perhaps I misheard. Did you say you were going to quantify the vegetation? And I don't remember you saying how you were going to do that.
Yes, I didn't include that unfortunately, but one of the ways is just biomass sampling. So like going through and cutting a specific area of the ground level cover. Also looking at tree canopy, diameter at breast height of the trees. Trying to think of what else. Yeah, those are generally what we'll be doing.
We might have time for one question, provided that it's short.
Follow up on just one of Alex's point about trees up in the flow country in northern Scotland, because of some quirks with tax law and stuff, they went in and planted forests in the flow country, which is just these blanket logs that just kind of droop over the landscape. And because the trees sucked up so much water, they were actually a net source of carbon. Even though you had these huge stands of trees, the best thing was to cut down all of the trees. So they're in the process of restoring these environments by deforesting a place that was afforested, so.
Okay, thank you very much, Anna. Speaker this afternoon is Carl Loewen. He started his undergrad at TU in 2020 and earned his honors degree in biology by May of 2024. His undergraduate thesis focused on developing a general environmental DNA technique, also known as eDNA, suitable for the Assiniboine River. With support from his undergraduate and master's supervisor, Dr. Barry Madison, his current research investigates a declining species in that river and explores the potential of their environmental DNA strategy to enhance conservation efforts. So tell us all about it, Carter.
All right, thanks. So, yeah, I actually requested that Dr. Shred not speak about my title. It's something I eventually probably should have because I got the date wrong on here, so we wouldn't have to look at the slide for quite as long. However, today we're going to look at a species known as the Mooneye in the Assiniboine River. Our thoughts on how on its potential to possibly be declining, and how we can look into that using an approach known as environmental DNA, or as I'll refer to it in presentation as DNA. So because this research was conducted in Manitoba and on traditional lands, it's important to first begin with land acknowledgements. So I'd first like to acknowledge that Brown University and the Assiniboine River are located on the traditional lands of the Assiniboine, Dakota, Anishinaabe, Cree, Oji-Cree, Dene, and Métis peoples, which use the Assiniboine River as a source of food, water, and recreation for generations. Hopping right into it, let's first look at a research question. So can we develop an assay that can be used to locate hydon, or in this case, just general Mooneye? So Mooneye are a species of this hydon genus, and other geographically relevant species towards those Mooneye in the dynamic Assiniboine River, furthering their conservation without harming that environment. And our ability to answer this comes down to eDNA. So let's first look into why use eDNA. So eDNA is incredibly cost efficient. Maybe not as cost efficient as angling, however, it's still a whole lot easier. It's very sensitive. It can be seen as sensitive as ddPCR in many scenarios. It's also extremely non-invasive. So when I say non-invasive, I mean we're not harming the environment by using a technique that's supposed to be saving the environment. So we're not actually removing or harming any pieces of that environment. We're simply taking a water sample and placing that water directly back into the watershed. Here's some of the alternatives. So one of our alternatives is gillnetic. This is how in the past these studies have been conducted, and it's an option that's not necessarily great. As you can see, every fish in that gill net is dead, so it's going to kill pretty much 100% of what you're sampling in a time that you're trying to be saving those species. Other alternatives involve electrofishing. So you can see here is this guy. He's got these probes stuck in the water. It's going to zap the water with electricity, and the fish are going to float up. Ideally not killing them, however, can cause some destruction. It's also insanely expensive. And then the other one is underwater visual census. So as you can see, this guy, he's got a pretty easy job here down in the water, doing his underwater visual census. Pretty easy to see the fish and water quality like that. However, our water in Brandon resembles chocolate milk, making underwater visual census quite difficult. So why here? So we're looking at the Assiniboine River in particular, and part of that reason is because this is where the Mooneye are, and I'll get to that in a second. However, why here is an interesting point. So as you can see here, some photos of two floods of the century from 2011 and 2014. So the belief is that this is, the wide belief around flooding, especially when you're having two floods of the century happening within a span of 5 years, is this a potential impact of climate change, altering some weather patterns. And if we can see that climate change is occurring, these floods are going to continuously happen. So in that case, how's this going to affect the fish and things like that? The other reason for why here is we have a tremendous amount of dams that completely or partially restrict water flow within our region. So the first being the Shellmouth Dam, which dams the Lake of the Prairies from the Assiniboine River near the Saskatchewan border. We also have a dam in Brandon, the 3rd Street Dam, which you can see from the 1st Street Bridge, which at times when the water is extremely low, maybe not even extremely low, slightly lower than usual, it can completely restrict anything from swimming through. And then finally, in the farthest point of our extent is the Portage Diversion and dam at Portage the Prairie, just about 3/4 of the way between Brandon and Winnipeg, which can restrict water flow there as well. So why these fish? So I'll get to a better distribution map here right away, but you can see that they're distributed quite sparsely. more so in the southeastern United States as well as pretty much Manitoba. So it's an area that we can look at as a possible area where we have a large amount of the world's population of this fish, maybe only living in our area. They're also ancient river monkeys, so they're the only, they're one of two extant species, the goldeye and the mooneye, which we both have here. When I say here in Manitoba, of the Osteoglossophoid family, so the bony-tongued fish. They're related to some South African or South American counterparts such as the arowana and arapaima. So if you've ever seen river monsters, those are the big ones that are on there, possibly eating people. However, that's not what we're looking at here. We're looking at fish that look like your ordinary fish. So if I asked everyone to picture a fish in their mind right now, they're going to picture a mooneye. They're very, very standard looking, with a nice silver eye on them, as you can see in this picture. So in terms of their habitat, they're much less common in turbid waters, and they're more common in slower moving rivers. So if I ask you to now think about the Assiniboine River, you're not thinking less turbid, turbidity meaning the murkiness of the water. You're thinking that's a pretty turbid water, it's brown. And it's a fairly quick moving river. However, in comparison to the Red River, say in Manitoba, it is actually moving quite a bit slowly. In terms of feeding, they're mid-water surface level feeders, and they feed on aquatic insect larvae, emerging flying insects, and emerald shiner. However, that emerald shiner piece is a bit lost in the literature, as it's never actually been proven, though it's cited, in so 40 different papers. The ecological role, this is the most important portion of the slide, is the mid-trophic level feeders. So what that means is they're prey to large predatory fish. So in the perspective of someone that's not looking into the mooneye, the important fish, like the channel catfish, walleye, and pike that everyone are hoping to catch when they're out fishing. They also feed on the smaller, like I just said, insects and emerald shiner. So this means that they can possibly be a bioindicator. They're that middle piece, the middle ground, that can show you if maybe those smaller species are doing poorly, these fish will also be doing poorly. There's nothing to feed on. If the larger fish are doing poorly, potentially they're showing that these fish are declining as well, as they have nothing to feed on. They're kind of that middle ground. And that's an aspect that we look to look into. Their human importance, so they're twin pretty much, goldeye, which is the same looking fish with the gold eye, are sold as smoked Winnipeg goldeye. In Winnipeg, we've actually named our only professional baseball team after them. So the mooneye are almost never distinguished from goldeye. So if you're buying smoked Winnipeg goldeye from a store, there's just a good chance you're actually buying mooneye. So their current conservation status. So this is an interesting map. So there's a lot to take away from it. So Manitoba here, globally actually, we'll get into that first. They're listed as G5 secure, so global 5 secure, national 5 secure, and subnational in Manitoba, subnational 5 secure. So that's interesting. However, this data is believed to be quite old. We can look into some other areas where the data may be more recent. So just when we began this project in 2022, New York here, which is now seen in orange as imperiled, was also this dark blue color. However, they began to study it just last year, and since then it's now become this S2 imperiled status, as well as some other states, Georgia, North Carolina, and Michigan, all in that S1 category actually. New York, Illinois, Arkansas, and Oklahoma, all in that S2. And finally, actually, of the province of Saskatchewan, our next door neighbor, classifies them as S3. So let's now look into why them and why here. So why the Assiniboine? Well, we first need to acknowledge that former professor here at Brandon University, Dr. C.L. Glenn, is actually the most recent person to study Mooneye in Canada. So when those studies were done in the 1970s, and he covered their seasonal diets, annual growth rates, parasitic infections, fecundity, things like that, of these fish. However, since then, the last paper being published in 1978, nothing since for an entire country. The Assiniboine River is also highly turbid, which is unlike the usual high habitat. However, this poses issues for the eDNA protocol itself. It kind of works against us from the get-go. However, if we can develop a protocol that will work here, it should work pretty much everywhere. And why here? in YBAM, the hydon could be used as a broader example of the activities in the environment, like you just covered with that mid-trophic level feeder aspect. And New York has designated them as now a species at risk. So NatureServe still has that F2 categorization. However, in their most recent papers that have now been published, they're now listed as either a species at risk or endangered. And American researchers have begun studies. So at some point, we need to follow suit and do the same. So eDNA sampling, what is it? So eDNA sampling is simply small genetic material that is sloughed off of fish, plants, insects, anything like that into the water that we can then go in, take a water sample and refine that DNA out of the water, giving us a presence absence signal for yes, that species is there, no, that species isn't there. So the uses for it are species detection, obviously. Determining that presence and absence. It can also be and has been used in invasive species monitoring. So at Clear Lake, the zebra mussels were found using this very technique, this eDNA technique, before anyone had actually ever seen them. It's also, again, a non-invasive technique. We don't want to be going in with a net and killing everything in the river when we're trying to save something that's in that same river. So we'll get quickly into the protocol that we've developed and are continuing to develop. So we'll look first at the field protocol and our field sampling mechanisms, and then kind of follow that up with more so of Caden's work in the lab, in our laboratory protocol and preparation. Finally, getting to through our purification, PCR, and our presence absence gel. So here's what we do. So we get to the water. We're going to take a scoop of this water, which ideally contains all of the DNA. Then we're going to run that through a filter that's inside of our peristaltic pump mechanism, which I'll show you a picture of right away. Moving over into our PCR. So if anyone doesn't know what PCR is, just think about it as your standard COVID test. It's going to give you, yes, you have COVID, no, you don't have COVID, but in this case, it's yes, there's fish swimming right beside you, or no, there's no fish swimming right beside you. And then into our gel, finally giving us that presence absence data. So our field setup. So this is what we take out into the field when we sample. This is our pump right here. So here's our water sample that is going to be funneled through our peristaltic pump. Within this filter cartridge is a replaceable disc, which is actually our filter, but the DNA will get trapped inside, and then we can take that back to the lab without actually having to transport any water away from the watershed. So this filter membrane has a 1 micrometer pore size, trapping all of that DNA that we're hoping to grab. Once we are finished, all of the water is passed through. We can then take that disc out, cool it, put it on ice, bring it back, and freeze it in the lab. So that sampling protocol that I mentioned. So once we get on site, we're going to take 2 samples, one upstream and one downstream of our sample area. And this is all going to be contained within one filter. So we're going to have 5 mini sites, 5 smaller sites that are separated by 5, 10, 15 meters based on the site and access points. And being able to contain that all within one filter gives us a presence absence data for a larger area than we would had we taken maybe 5 samples in different spots. Then we're going to return to the lab, digest that filter, and conduct our PCR with the primer sets that I'm going to talk to you about. Finally, again, giving us that presence absence data. So our DNA purification with that filter that we're taking back to the lab, we can add lysis buffers, swash buffers, elution buffers to try and rid of some of those inhibitors that may be in the water, making it more difficult to isolate that DNA from the filter. Washing it, finally eluting our final DNA that we use for PCR. The other thing that we need in PCR is primers. So what primers are is they stick to the DNA and they make multipliable copies of that DNA strand so that you can have that presence absence data. So, however, primers need to be developed for each individual species. So the one that we're most focused on is hydon, obviously, our Mooneye. Our Mooneye and goldeye both fall under this hydon umbrella term. However, we've also tissue validated some other species: channel catfish, stonecat, walleye, yellow perch, and shorthead red horse, to be able to get a broader sense for their trophic role in the future. This is an interesting one here, this My Fish primer set. So this MyFish primer set is actually a primer set that will test present or absent for any fish that's in our local watershed. So it'll say yes fish or no fish, but it won't break it down further than that for us. We also have a ton of unvalidated primer sets, which we are awaiting tissue to be able to do some tissue validation studies on to ensure that they work. So what does that tissue validation look like? So here we have our Hydon, Mooneye, Goldeye, and our MyFish primer sets. So in this first lane here, we have Mooneye tissue with our Mooneye primer sets, and I apologize for how oversaturated this image is. However, this is our presence for Mooneye DNA within that sample. Second lane, we have Goldeye tissue and Goldeye primer sets. So again, present. Third, Hydon primer sets. So this will test positive for both Goldeye and Mooneye, giving us a general Hydon primer set. Obviously positive with our Mooneye tissue. And finally, our MyFish primer set. So this is supposed to test positive again for every single fish species, and it had. So when we get back, so once we have these primers validated and ready to go, we can then conduct our PCR analysis using those primer sets, the polymerase and the purified DNA, all three things that I've just talked about for our PCR, which will then give us again that presence absence data. Oh, I'll get back to that. So what's been found? eDNA results. So this is what we get back from a field sampling day. So this test was conducted on, or this sample was gotten on July 19th of this year at the City Brand Water Treatment Plant at the end of 26th Street within the river, obviously. And we can see on this gel, although very faintly, that we have presence for two fish species. So in lane 9, the channel catfish, and lane 10, that MyFish primer set, which can work as a positive control if we have another fish that tests, if we have a species that tests positive, we have that positive control that we also know that the fish is testing positive. Also, very a lot of lanes with absence, so that means that all of these species here were not found in this exact study. So if we look at another sample day, on this sample day, we actually found Mooneye. So we can see here, doesn't come through very well. However, there is a band there testing positive for our Mooneye. And finally, at the end, this is our genus level Hydon primer. So it also tested positive. Then September 19th, 2023, we have our this is an interesting one, so I just threw this in here for interest's sake. So this day we had one presence, and it was only that general fish primer. However, none of the other primers tested positive, meaning that it's a primer set that we didn't use that exact day, or possibly not tissue validated, or possibly that we don't have something that something else is in the water. So where does this leave us? So Hydon are species at risk in similar environments. So other environments like New York, like I just spoke about, where this has been studied, have these fish designated as species at risk. Very similar environment, so that's a possibility that has also come to here. We've also located these Hydon, just now needing to analyze what that means for their trophic level, or for their trophic level, and how they're doing. Interestingly enough, Manitoba still allows the use of these fish species as bait. So if it is a disappearing species, it's very important to get past that point, get people aware that these are struggling species, get those laws changed so that that's no longer a thing that we can do, as this is obviously leading to way higher mortality than it needs to. In terms of our protocol, we now know that field sampling is viable. We have presence and absence data from all of these sites. Now, and we know that this idea has no environmental impact. With that Hydon confirmation, we can now say that we found fish without seeing fish, which in my opinion is a pretty cool aspect to our research. So where do we go from here? So for Mooneye in particular, we have just received our scientific collection permit, which will now allow us to collect species to analyze stomach contents, that kind of thing. To be able to see, first of all, potentially prove that they eat those animals shining. However, we can also look at this in the broader aspect where we can also collect some channel catfish, be able to see that they're eating Mooneye. So one thing that I haven't mentioned yet is Mooneye are migratory species, so they're moving through this area constantly. However, kind of tough to do an eDNA study if they're migratory and they just happen to not be here. So we can analyze those catfish that we can now, which eat these Mooneye, we can analyze their stomach contents to be able to understand when they're here, which will give us a secondary opinion to our eDNA. This will allow us to analyze their trophic role, both of what they eat and who eats them and when. And we will be able to do this research along with some kind people at Manitoba Fisheries who have reached out hoping to work with us. In terms of eDNA, where we need to do some various things such as a lunar detection study. We need to know how much DNA we need to be able to get a positive result. This will give us ideas such as maybe we don't need 15 pounds of DNA to be fully passed to give us presence, or maybe we do need a little bit more than we had initially thought. We also need to analyze the effects of those inhibitors. So turbidity, first and foremost, gets stuck in our filter. It's a pain. It's dirt. So it kind of hurts our abilities to be able to process the water through our pump. Also, things like humic acids and other things that break down that DNA can have significant effects, obviously, on a DNA study. Then our degradation rates. We need to be able to understand how quickly that DNA is degrading once we have it or once we're transporting it, that type of thing. And finally, cadence velocity and cases that hopefully you guys were all present for last Monday at the biology talks. So a couple of acknowledgements. I'd like to acknowledge Supervisor Dr. Madison, committee members Dr. Rutherford and Cushon, Caden for all of his work in the lab, Stephen Keon for donating us some fish tissue, our field studies of biology course, various NSERC discovery grants, and I actually took Dr. Mel's name off this list today because the last two times I acknowledged him, he didn't show up for the presentation. So he's been removed. I'll put him back on for today, though. He's been very helpful in the lab, teaching me how to work that type of thing.
Your father wouldn't be here.
Thanks for those things. I did.
Okay, thank you very much. Some questions? Yes.
Just infrastructural, I guess, is it possible to somehow have a pre-filter unit that will get rid of your turbidity, but not your DNA?
Yes and no. There's actually probably simpler ways to do that, using a pre-filter. So having something with wider pores to allow lots of that dirt to get stuck in there. So we have done that, and that's another thing with those rates that I was talking about earlier is we can do a study again with that pre-filter to ensure that we're getting that same signal and none of that DNA is actually getting stuck within that pre-filter.
So maybe one more question, that's on time.
Do you do Sanger sequencing on your positive fans to ensure there's no false positives?
We haven't, no. But obviously that would be a very good idea, something to do down the road. Up until this point, it's been based on getting that presence signal, hasn't been overly easy. So the ability to now that we have that ability, we know that we can get that presence or absence data. Now we can get into things like that. That we can include in a future plan for sure.
Is it like ultimate, sorry, like one more question. Is the ultimate goal to use it like in combination with something like digital drop PCR or assessing both presence and prevalence?
Yes and no. The eDNA portion of this, I would say has more implications in solely that presence absence. And now that we know they're here, I guess we can go into those things. However,
is there any benefit to going into that, knowing if there's actual numbers or?
Well, absolutely, there's benefit in knowing how many are there. I'd say that there's more benefit in understanding why they're there and where they are, to be able to promote that conservation in the future rather than just knowing that there's a bunch in this pocket doesn't really impact anywhere else, right? So maybe if they, like, you can have huge abundance in one area, but still have an endangered species overall. So I'd say that that's a more broad goal for this project.
Thanks, very much.
Okay, thank you very much. Okay, so our next presenter today is Kieran Taylor. He's obviously a graduate student in the MELS program. He earned his BSc honors at the U, majoring in biology and double minoring in both chemistry and mathematics. He completed an honors thesis at BU during his undergrad, where he developed a qPCR-based diagnostic test to test for the soybean pathogens Cercospora Fuchsii. So Kieran today is going to be presenting his current work in the MELS program, where he hopes to answer the following question, which also happens to be his talk title. Do fungi impact disease resistance in barley? So looking forward to hearing the answer.
Great. Well, thank you very much for the intro. Greatly appreciated. And yeah, as Dr. Schreck just said, I'm just going to be giving a brief presentation of the research I've done, seeing whether or not certain types of fungi impact disease resistance in barley. To begin, let's talk about barley itself. Barley is one of the top 10 most commonly grown crops worldwide. And most barley, or at the very least a good chunk of barley in Canada, is going to be grown for malting purposes. This means that a large portion of barley is going to be grown to produce things like beer, Scotch, and Irish whiskey. However, while about half of all barley is grown for malting purposes, a roughly equal portion of it is also grown for use as animal feed. So even if you're not exactly a fan of alcoholic beverages, barley is still a very important crop if you want to enjoy things like burgers or bacon. In addition to that, a small portion of barley is also grown for food. Now, barley is a very important crop to us here in Canada, mainly because Canada is the 5th largest producer of barley in the world. In fact, in 2023 alone, we produced over 8.9 million tons of barley by seeding about 3 million hectares of land. So as you could tell, barley is quite a prevalently grown crop here in Canada. However, barley is also important to us economically. In 2022, for instance, barley production basically added over $14.4 billion to the Canadian economy. During that same year, barley production generated over 100,000 full-time equivalent jobs, which generated over $6.1 billion in wages. So obviously, barley is a very important crop to us, not just because of the products we can make from it, but also because of the economic value it has to us in Canada. However, like with all crops, barley is susceptible to disease. In particular, the disease that we are looking at in our studies is what's known as Fusarium. Now, Fusarium hepatitis is a very nasty disease which produces a toxin in barley known as deoxynivalenol, the structure of which is shown. Notable about this toxin is that as little as 0.5 milligrams of this toxin is all it takes to ruin a full kilogram's worth of barley. Mainly because if we find that in a kilogram of barley, it can no longer be used for malting. As I mentioned before, about half of all the barley produced in Canada is used for malting. And so if you can no longer use it for that, it's obviously technical losses for farmers. And to put that 0.5 milligram number into perspective, that's as heavy as 10 grains of sand. That is how little of this toxin it takes to ruin a full kilogram's worth of barley. So obviously, these sorts of crop diseases can cause massive financial losses, and we would therefore want to basically mitigate. The only issue is trying to usually mitigate these with fungicides. And the problem with that is that fungicides can often be expensive. It can sometimes be difficult for farmers to coordinate when they should spray their field with fungicides because poor weather can prevent them from doing that. In addition to that, fungicides can cause damage both to the environment and potentially to human health. So frankly, the less we have to use fungicides, the better. The only issue is when these sorts of diseases can cause such significant damage to crops, how do you avoid using fungicides? That's where my research comes into play. I'm studying a type of fungi known as arbuscular mycorrhizal fungi. Now, for the majority of this presentation, I'm just going to refer to them as these fungi, mainly mycorrhizal fungi over and over again, would frankly get a bit tedious over time. So just know whenever I say these fungi, I'm referring specifically to arbuscular mycorrhizal fungi. So what is so special about these fungi? Well, These fungi are capable of colonizing the roots of plants with fungal structures such as hyphae, and they also form branching structures within the roots of plants known as arbuscules, labeled with an A in this diagram. The reason they do this, for plants is that when they colonize the roots of plants, they can provide plants with extra soil nutrients like phosphorus. As you can kind of see in this diagram here, the hyphae of the fungi
So for my research, I wanted to answer two particular questions. Firstly, do these fungi provide
disease resistance to barley, and in particular, disease resistance to Fusarium pet blight, which is shown in this spike of barley here. So do they provide disease resistance to barley? And if so, do certain species of these fungi or do certain levels of these fungi provide greater, basically, does the species of fungi colonizing the roots of the plant matter when it comes to providing disease resistance? And does the level of fungal colonization on the roots also matter when it comes to disease resistance? These are the questions I wanted to answer with my research, and to do so, multiple different genotypes of barley, or like different genetic varieties of barley that have known differences in disease resistance. So for my study, I'm looking at 4 different genotypes of barley, these being CDC Bold, AAC Synergy, AAC Connect, and Kutaya. And as you can see in this diagram, I've listed them here in order of these to most resistant. When I say these, I mean specifically Fusarium pet blight. So, since we want to know whether or not these fungi provide significant disease resistance to barley, we want to gauge whether or not these fungi are in disease resistance that we see these different genotypes. Now, in order to tell whether or not they actually are contributing to that difference in disease resistance, we would need for there to be some sort of difference in the type of fungi colonizing each genotype. Because think of it this way. If all 4 of these different genotypes had the exact same levels of fungi and the exact same species of fungi associated with their roots, there's no good reason to suggest that those fungi are contributing to the difference in disease resistance that we see across these different cultivars. So basically, in short, you can really only conclude that these fungi are contributing to the differences in disease resistance across these genotypes, differences in the fungi associated with each genotype. With all that being said, our hypothesis right now is that these fungi do indeed provide significant levels of disease resistance to barley. However, for this to be the case, what we should expect to see is something like this. So what we would expect to see is that there would be particular species of fungi that are associated with the more resistant cultivars or genotypes of barley that are not found on the less resistant genotypes. So in this diagram here, I've basically labeled specific fungi that designate that they are found more resistant genotypes of barley. And as you'll notice, none of those blue species are found on the least resistant genotype CDC Bold. This would be an indicator that those particular fungi species are likely playing a significant role in providing disease resistance to these more resistant genotypes. And so this is something we might expect to see if these fungi do provide disease resistance to barley. Similarly, if they provide disease resistance to barley, we would also expect that there would be a difference in the level of fungal colonization on the roots of plants based on their level of disease resistance, where the more resistant genotypes of barley would have more fungal colonization on their roots compared to the less resistant genotypes. And with regards to both of these sorts of expectations, there have been some studies that have shown that specific species of fungi do provide greater levels of disease resistance than others. And there is at least one study that I found that seems to indicate that the level of fungal colonization does contribute to the level of disease resistance provided by these fungi. So with that in mind, that is why we're going to be looking for these sorts of things in our different genotypes. Now, to answer, therefore, whether or not these fungi provide significant disease resistance to barley, we need to figure out the level of fungal colonization on the roots and which species of fungi are on the roots of these 4 genotypes. To determine the level of fungi on the roots of these plants, we're going to use what's known as the magnified intersections method developed by Dr. MccDonald. To use this method, what we first do is we take our root system from the barley and we cut it up into 1 centimeter long fragments. We then take these fragments and we stain them with a black stain in order to basically show the fungal structures embedded in the roots under the microscope more clearly. After this is done, we then line up our root fragments along the length of the microscope, and at specific set points across the microscope, we make observations of our roots. If there are root fragments located at these specific set points, we will examine those root fragments under the microscope to see whether or not there are any fungal structures embedded within the roots. And we will continue to examine our root fragments at specific set points in this way until we've made our way across the entirety of the microscope slide. Now, in this here is just a crude diagram of what you would kind of see when you're looking at one of these roots under the microscope. These white boxes represent the individual root cells. These black structures here represent the different fungal structures you could see under the microscope, whether that be hyphae, vesicles, or arbuscules. And these dotted lines represent the different set points in which we are observing the root under the microscope. Now, as you can see in this diagram here, there are 5 different observations we've made of our root system. Of these 5 observations, 4 of them have hyphae in them. That indicates that 80% of our root system is colonized by hyphae. Similarly, one of the 5 observations we've made has a vesicle in it, indicating that 20% of our root system in this example is colonized by vesicles. And lastly, one of the 5 observations has an arbuscule in it, indicating that 20% of our root system is colonized by arbuscules. And so you can kind of see that by using this method, we can gauge the level of fungal colonization in the roots under the microscope. And therefore, we can distinguish whether or not different genotypes of barley have different levels of fungal colonization on their roots. Now, I should note that for this study, we are primarily going to focus on the arbuscular colonization. The reason for that is that basically there are different species of fungi that can sometimes associate with the roots of plants that are not arbuscular mycorrhizal fungi. This means that seeing hyphae and vesicles under the microscope is not a guarantee that there's actually arbuscular mycorrhizal fungi on the roots of our barley. However, as the name arbuscular mycorrhizal fungi implies, arbuscules are characteristic of these types of fungi. And therefore, by seeing those under the microscope, we can get a much more accurate, if albeit a bit more of a conservative estimate as to the level of fungal colonization under the roots of the microscope of the specific types of fungi that we are looking for. And so that's why we're going to be using the arbuscular colonization primarily to gauge the level of fungal colonization on the roots. So that's how we'll gauge the level of fungal colonization, but what about the species of fungi on the roots of the plants? Well, to figure out which species of fungi are on the plant roots, what we'll have to do is extract the DNA from extract the fungal DNA from the roots of the barley, and subsequently we will amplify the DNA using PCR. We'll do this so that we have enough DNA to work with, but it will also be done so that we can gauge the relative abundance of different fungi species later on. Because we'll think about it this way. Let's say we take our fungal DNA sample and we amplify it with PCR so we get multiple copies of the DNA we were working with. And let's say following this, about 90% of that duplicated DNA is associated with, say, one particular fungi species. That would indicate that that was likely much more prevalent on the roots of that genotype compared to any of the other fungi species. So by looking at the relative abundance of different fungi gene sequences associated with a particular species, we can determine the relative abundance of that fungi species on the roots of each genotype. After amplifying the DNA using PCR, we will CR the next generation sequencing. Doing so, we can then use these DNA sequences in order to determine which particular fungi species these genes and therefore we could then compare the different the different barley genotypes and see which fungi species are associated with each of the different genotypes. And so that's how we can therefore compare that. Now, if our hypothesis is true that these fungi do indeed provide significant disease-resistant disease,
Expect to see something like this. In this
we have four bars representing our four different barley genotypes. And here we're seeing the level of our vascular colonization. This arrow at the bottom here is just to signify the level of disease resistance, where the further to the right the genotype is, the more disease resistant it is. Now, if these fungi do actually provide significant disease resistance to barley, we might expect that the more resistant genotypes of barley would have greater levels of fungal colonization on their roots compared to the less resistant genotypes. And this would basically signify that the colonization levels of the fungi are contributing to the level of disease resistance that we see these fungi providing to the different barley genotypes. And so this would help to demonstrate that these fungi are contributing to disease resistance in barley. Similarly, in the event our hypothesis is true, we might also expect to see something like this. Here we once again have four different bars for our four different genotypes. And here we're seeing basically the relative abundance of certain fungi species on the roots of each genotype. Once again, the further to the right the genotype is, the more resistant to disease it is. If there are particular fungi species that provide greater levels of resistance than others, what we might expect to see is that the more resistant genotypes of barley would have specific fungi species associated with them that are absent on the less resistant genotypes. In this case, the red and blue fungi species would be associated with the more resistant genotypes and not found on the less resistant genotypes. This would demonstrate that those particular fungi species are likely contributing greater amounts of disease resistance compared to the other fungi species associated with the roots of the barley. And that would therefore help to demonstrate that these fungi do provide significant disease resistance to barley. Now, I do actually have some preliminary results to share so far. I've looked at about 20% of our barley samples under the microscope, and so far this is what we found regarding the level of our vascular colonization. Now, I will fully admit this does not perfectly align with our expected results. However, what I have noticed is that there seems to be a slight trend where AAC Connect, one of our more resistant genotypes, does have more arbuscular colonization than CDC Bold. However, as you can kind of see from the overlapping error bars, it is not a statistically significant difference. And so even though it's not statistically significant, there seems to be a mild trend. And like I said, this is what I got from looking at 20% of my data. I still need to analyze the other 80% to confirm whether or not there actually is a trend relating the level of fungal colonization to the level of disease resistance provided by these fungi. Now, I just want to finish by basically discussing some of the benefits our research could have. Because if our hypothesis is true that these fungi do provide significant disease resistance to barley, this could be very useful information both for farmers and for those developing new varieties of barley. For farmers, obviously, if these fungi provide disease resistance, farmers could potentially use these fungi in their fields to mitigate disease in barley by potentially, say, inoculating their fields with these fungi, and thereby these fungi could colonize the barley, provide them with disease resistance, and in doing so, that could potentially mitigate the use of fungicides in farming, reducing the environmental damage they can do. Additionally, this could be very important information for barley breeders, because if we know for a fact that certain species of fungi or certain levels of fungi are associated with disease resistance, well, barley breeders could take that into account to develop new varieties of barley. And if we find that these new varieties of barley attract those particular fungi species, well, that could help to basically amplify the level of disease resistance in these new varieties. With that being said, I would like to thank the Brewing and Malting Barley Research Institute for their funding. I'd like to thank my supervisors, Dr. Anna Badia and Dr. McGonigal, for laying much of the groundwork of this research. And I would like to thank Dr. James Tucker for his work on the advisory committee of this research. I would also like to thank everyone else in the barley breeding program who helped me get all the samples that I needed for this research. And last but certainly not least, I would like to thank all of you for listening.
Thank you very much, Karen. So we've got about 4 or 5 minutes for questions.
Yes.
So assuming when you run the other 80%, you do get more of your uptick and it sort of works as predicted.
Yes.
Go to like fungirs.com and buy this to spread it, or can you cultivate, like how do you?
This would kind of just depend on which species we end up finding. The issue with these fungi is that they're referred to as like obligate symbionts, meaning they to be associated with particular species of plants. So generally, yeah, we would probably have to go to some company that's already selling this, or alternatively, we did obtain some of the samples that we already have with fungal spores or something like that to use as inoculum labeling. That's assuming that we do use inoculation in certain studies. You could also potentially, because this is one thing that I maybe didn't emphasize as much, we are specifically looking at fungi species that are already present in the soil. So these are fungi species that are already present in the soil, and we're seeing the form associations with specific fungi already there much more prevalently. So it's possible that some of these species are already found in the soil, and by doing certain farming practices like avoiding tilling, you could potentially amplify the level of those fungi already.
What's the mechanism for, is it just more water and nutrients, therefore healthier, or is there something else going on that, fungus?
That is a great question. There's like 4 different theories on how it works.
Okay.
I think
in theory is that they find specific, basically disease resistance associated genes in the plants, so that they effectively, the way I would almost compare it is kind of like how a vaccine allows you to respond much more quickly to a disease because you've already been kind of exposed to it. In that same way, these fungi are kind of, at least this is believed, this is not a guarantee of how the mechanism works. But the idea is that these
fungi, when they associate with plants, find disease resistance related genes such that they can effectively produce the proteins and such needed to fight disease much more quickly and much more powerfully as a plant doesn't have this fungi associated.
Oh, one more question. Yeah, just what you're making, or you're deriving, what level of priority is disease resistance associated with that? Maybe you can say growth rates and things like that.
I'm not entirely sure as to what the hierarchy of priorities would be. Obviously, we are definitely going to try to prioritize things like growth, yield, and depending on it, we're trying to make a specific variety for molting. You might also prioritize the ratio of starch and stuff. But disease resistance is still a very high priority, especially because, well, the disease that we're specifically looking at, Fusarium headlight, is unfortunately, well, not exceedingly prevalent in Manitoba. It's still bad enough that we really want to avoid it whenever possible. So if we can find new varieties to combat that, we're obviously going to strive to do so.
Can it still be used for feed if it's contaminated, or is it just a malting that it can't be used for?
I think it can still be used for feed, but okay, yes, it can still be used for feed. I obviously don't, I think if it's in massive amounts, it probably, honestly, if it's a little too much for humans, it might still be enough for cattle.
Thank you very much. Okay, so next presenter today is Augustine Chuukwunta. He obtained his BSc in plant science and biotechnology at the University of Nigeria, where he carried out a thesis on the biosynthesis of silver nanoparticles using tropical medicinal plants and evaluated their antimicrobial properties on some bacterial and fungal species under the supervision of Dr. Obi Ugandu. So Augustine's talk today is entitled The Role of Bioinformatics in Plant Pathology. Go for it.
Thanks, Rick, and thank you all for coming. So today I'll be sharing what bioinformatics is why bioinformatics is important and how we can harness bioinformatics in controlling plant pests and pathogens. So bioinformatics is an interdisciplinary field that combines computer science, biology, and mathematics to solve biological problems. That is, in bioinformatics, we simply use computer with a set of mathematical algorithms to analyze complex biological data in order to solve biological problems. We could also define bioinformatics as a science that stores biological data in a database. We retrieve this data from the database and analyze this data using various bioinformatics tools and approaches. So bioinformatics is divided into two main subfields, which is development of computational tools and using these tools to better understand living systems. So bioinformatics tool comprises database and software. Database is an online system where we store biological data, while software are a set of mathematical algorithms that we use to analyze and interpret data. So in bioinformatics, the type of data that you are working with will determine the approach you will take in analyzing the data. So we have some key approaches in bioinformatics, which are gene expression analysis. Here we tend to look at the activity level of gene. Given, for example, you have a biological system that is infected with a disease. So you want to see how that biological system will respond to the disease, or when the biological system is being treated, you want to look at how the biological system responds to the treatment. Another approach is transcriptome assembly. So we use bioinformatics to study the entire genetic makeup of an organism, most especially a newly discovered organism, so that we can understand their biology, growth, and survival. So the third approach of bioinformatics, which is metagenomics, here we use bioinformatics to study the microbial community present in a particular biological system, so that we can see how this microbial community influences the growth and development of that biological system. So how powerful is bioinformatics? So with bioinformatics, we can predict and prevent diseases before they occur, and we can grow crops that are pest resistant naturally. So, and the most interesting thing is that with bioinformatics, we can achieve this faster and more accurate than ever before. So bioinformatics is like having a superpowered microscope that looks into the DNA of a particular biology system, let's say plants, and tells you exactly what you need to do in order to grow a perfect plant. So can we achieve all this without bioinformatics? Absolutely yes. However, it takes considerable amounts of time and energy. Take, for instance, you walk into a massive library with millions of books, and you are interested in a particular book, and the library does not have any catalog system. So you can imagine how long it will take for you to actually find the book that you're actually looking for. Or say you are a farmer, you are interested in growing a perfect tomato, one that is juicy and pest resistant. So first, you have to cultivate several hundreds of varieties of tomato. So you wait for them to grow. You test and see which one is juicy and which one is pest resistant. Then you try this over and over again until you get the perfect tomato. But with bioinformatics, we can simply analyze several thousands of tomato varieties in a fraction of time. So identify which genes that are responsible for juiciness and which genes that are responsible for pest resistance. Then we can predict which combination of genes to use in order to grow a perfect tomato. So, and the big challenge now is by 2050, the world population will reach a staggering 9.7 billion people. And the question is, how do we produce enough food for everyone? Some of the solutions were to make farming system more efficient, to improve crop breeding, and to reduce food waste caused by pests and pathogens. So for the purpose of this study, we'll be focusing on how we can use bioinformatics to reduce food waste caused by pests and pathogens. But first, we need to understand pests, pathogens, and the disease mechanism in plants. So for any disease to occur in a plant system, there has to be a favorable host, a virulent pathogen, and a favorable environment. So when these diseases do occur, we look at what caused the disease, the factors that are responsible for the disease, how plants respond to the disease, then the control measures. So why pathogens and pests? Up to 40% of crop production are lost to plant pathogens and pests. Each year, plant disease costs the global economy $220 billion. So these are huge problems that require urgent solutions. And the good news is bioinformatics is already helping scientists to develop better crops that are pest resistant. So in my thesis, I'll be looking at 3 different approaches we can adopt in order to control plant pests and pathogens. So the first approach is gene expression analysis of infected soybean. So here we look at the soybean infected with a fungus Alternaria. So we tend to look at how this soybean responds to the disease. Then we identify the active genes which are upregulated. Then we can also identify active genes in Alternaria so that we can target this gene as a pest control mechanism. So in the second approach of bioinformatics, I'll be looking at the transcriptome assembly where I have to study the entire genetic makeup of the 2 most prevalent fireworms in Manitoba, so we can understand their biological and survival. So in the third approach, I'll be looking at metagenomics. It's another approach of bioinformatics where we look at the impact of fungicides on Barley microbiome and to what extent does these fungicides have on microbiome, if they actually have any effect. So in the first approach, why soybean? Soybean are major source of food for humans as well as for animals. In the last 15 years, soybean has become an important crop in Canada, but there's a challenge. Alternaria automata is a fungal disease that actually destroys soybean. It causes Alternaria leaf spots. As a result, it causes stunted growth, thereby reducing crop yield. So because of this, farmers spend millions on fungicides every year, and yet the problem still, the losses keep piling up. So my research question is, how do pathogens like Alternaria alter the gene expression in soybean tissues? And my hypothesis is Alternaria infection triggers differential expression of key defense gene impacting soil-based resistance mechanism and yield. And my objectives are to identify the important gene in soybean and Alternaria, then knock down important genes in Alternaria as pest control mechanism. So for a given system like plants, such as soybean, so when soybean is infected with a disease like Alternaria, so it undergoes stress. As a result, the cells tend to behave differently from that of the healthy soybean. So then what we do is to measure the differences in the gene expression between the healthy soybean and the infected soybean. So to achieve this, we adopt 3 major processes. So the first is preparing sequencing library, which is to isolate the DNA from the plant. So the second step, which is sequencing. So we are going to arrange this DNA in a format in which you can actually interpret it. Then the last step is data analysis, whereby we will tend to see exactly what is happening with the infected soybean. So then this is more like the procedure. So we extract the RNA. Since we are expressing the gene expression, we isolate the messenger RNA. So we make several copies to have a stable cDNA. Then we add adapters. Then before we pass it into a sequencer, then the sequencing machine produces reads. So reads are just short nucleotide sequences of a living system of interest, in our case, which is soybean, that contains the genetic information of the soybean. So what we then do next is we map the reads to the reference genome. So here we'll be mapping it to the soybean reference genome, which we can easily obtain in the database like NCBI or SMBO to produce a quantification matrix. This quantification matrix is just telling us the activity level of the gene. Then we'll perform further downstream analysis to identify the genes that are actually active, which is upregulated, and the genes that are downregulated, which are non-active. So we have some results. As you can see, the orange are the cells of the healthy soybean, the blue are the cells of infected soybean. So as you can see that the blue is slightly separated from the orange, which shows that the cells behave differently as a result of infection. So the second result being that is the gene expression differences between healthy soybean and infected soybean. So the orange gradients represent upregulated genes, that is genes that are active, while the blue gradients represent genes that are not active, that is downregulated. So as we can see, the first 6 genes here, so we see that they are downregulated, and the last 4 genes, as you can see, yeah, so they are upregulated. So these are bioinformatics tools that we use to achieve our objective, which is to identify upregulated genes in both soybean and alternaria so that we can target the active gene in alternaria for as a pest control mechanism. So let's shift our focus now to the second approach of bioinformatics, which is transcriptome assembly of wireworms. So wireworms are soil-dwelling larvae of click beetle. They feed on seedlings, roots, and below-ground crops. Wireworms are interesting pests such that they have a very long lifespan. They can spend up to 5 years in soil, and they damage up to 60% of crop yield. So the problem is, presently now, there are no approved control measures to control wireworms. So in 2004, the government of Canada banned the use of Linden, which is a pesticide for controlling wireworms. So as a result, there has been a dramatic increase in the spread of wireworm in Western Canada. So Manitoba happens to be the ground where we have the majority of these wireworms. And the 2 most important wireworms that are causing the great havoc in Manitoba are the H. abbreviatus and L. californicus. So before we manage this important pests, so it's good for us to understand their life cycle. So the click beetles, they overwinter in the soil, and in spring they emerge. The adult click beetle, female click beetle, lays between 200 to 400 eggs. So by summer, the egg hatches, and then we have different instar stages. So from the first stage to the fourth stage, it takes actually 5 years, up to 5 years in the soil. So at this stage, it causes great havoc to plants. So they are also regarded as the most destructive phase in the life cycle of click beetle. However, the adult does not pose any significant threats to plants. So my research question is, what are the key genes involved in the growth and survival of wireworms? My hypothesis, wireworms have a unique set of genes that are activated to negate their survival under stress. My objective is to identify key functional genes in wireworms to target for pest control. So these are the different stages of wireworms. So we are interested in identifying the key genes at each instar stages, determine the instar stage that is causing the greater impact, then compare key functional genes across all the instar stages for pest control. So just like the differential gene expression in soybean, so we are also adopting the same 3 processes with wireworm. So these are the tools we use to achieve our objective, which is to identify the key functional genes in wireworm so that we can target as pest control strategy. So we'll be shifting our focus now to the third approach of bioinformatics, which is metagenomics of Barley microbiome. So metagenomics is a science that studies the entire genetic material that is present in a particular environment. So microbiomes are a collection of microorganisms such as bacteria, fungi, and viruses. So when we say microbiome is like a tiny, invisible ecosystem that helps us to maintain health and balance. So for instance, the human digestive system has billions of microbes that helps to digest food to produce energy. So depending on the type of food or diet that you eat will determine the activity levels of the microbes in the digestive system. So now, why should we care about microbiome? So in 2001, Dr. Terry Walsh was diagnosed of multiple sclerosis. So by 2007, she has declined to using a wheelchair. So after 1 year of changing her diet, so she was back on her feet. Also, the microbiome of healthy mouse was transfused into that of unhealthy mouse. So after some time, the unhealthy mouse started showing some stress-related symptoms. Similarly, an experiment was conducted on fruit fly. So there was a certain bacteria in the gut of fruit fly that digests protein. After the microbiome was changed, the fruit fly started feeding on sugar. So just like the microbiome in animals and plants, in animals and humans, plants have their microbiomes in leaves, fruit, and soil, and it helps to absorb essential nutrients and protect them from harmful pathogens and diseases, and even boost growth. But the key challenge here is fungicides actually kill this microbiome. So my research question is, how do fungicides impact the composition and diversity of beneficial microbial communities in Barley? And my hypothesis is fungicides disrupt the Barley microbiome by reducing the diversity of beneficial microbes, thereby impacting growth and resilience. So my objectives are to characterize the microbial community in Barley and to determine whether fungicides impact Barley microbiome and to what extent. So here, I'll be collecting the microbiome in the leaf, spike, and roots of Barley. Then I will check whether fungicides reduces or increases the microbial activity in this part of Barley. Then I will determine how the changes in the microbial community as a result of fungicides application impact Barley health, such as disease resistance, nutrient uptake, and growth. So this methodology of collecting the microbiome from Barley plants. So we extract the DNA. So just like the soybean differential analysis, we sequenced it. Then we classify this DNA to which organism they belong to, whether they belong to which class of microbiome, microbial community, whether it's bacteria, fungi, or virus. Then we do taxonomical profiling so that we can understand the function and structure of these microbes and the influence they have on the plant. So these are the bioinformatics tools that we use to achieve our objective, which is to see whether fungicides influences the microbiome in Barley, and if they do, to what extent. So in summary, so my project basically is looking at 3 different approaches of bioinformatics. The first, which is the gene expression analysis of soybean, which is to identify the active genes in soybean and alternaria, so that we can target this active gene in alternaria for pest control. So my second approach, which is transcriptome assembly of wireworms, that is, I'm trying to identify the key genes in wireworms, so that we can target that as pest control mechanism. And the last approach, which is metagenomics analysis of Barley microbiome. So I'm looking at the impact of fungicide application of Barley microbiome, and if they do actually have an impact, to what extent. So I would like to thank my supervisor, Dr. Kasoni, my research committee, Dr. Adeli and Dr. Lamoy. I also like to thank Dr. James for helping us with the Barley samples, and my colleague, Toby, as well as NSAC, for funding this project. Thank you all for listening.
So coffee might be here. So hopefully you can split your time between either getting coffee or asking questions.
Are you able to go back to your gene expression heat map from the soybean? Sorry.
So lots of animation.
Sorry.
Yeah.
I was just wondering like what specific genes that, oh yeah. Or sorry, maybe I'm misunderstanding, but what is the specific genes that you're looking at there, or what is GLYMA? I'm not.
So for now, once we do the analysis, so we identify the active genes. So we are not sure of the particular gene of interest. So then in the active genes, we look at those ones that are more expressive. Then we can then further analysis, which is functional analysis. So we try to look at the function of those genes and how that gene actually affects soybean.
Okay,
Then in future, we can actually target that.
okay, thank you.
Other questions?
Okay.
It looks like 3 projects, about the work. Are you going to do all are you just focused on the computer-based work, or are you doing laboratory work with the DNA as well?
So if you can say that again.
Are you actually in the lab with the pipette, or are you just doing the computer analysis?
Yeah, so basically I'm doing the computer analysis, but the final step, I'm also doing the lab work, which is the wet work, as well as the using the bioinformatics tools to interpret and get some insight.
So for, you basically have like 3 pipelines for the 3 parts of your project. And they all start with like FastQ and then Sherman.
Yeah.
And then they differ from there, even though they're kind of similar steps at other points. How do you decide, you know, which algorithm or software to use for similar steps, but for different projects?
Yeah, so so we have some works that have been published. So it's just like trying to get insights from the published work. So looking at what tools they use, then by your own spare time, you research on those tools and see how you can take advantage of the tools in your own particular research. So as you can see, we have at the beginning similar tools with all the approaches. Then after the 3rd step, so the tool starts changing. So depending on the type of data that you have, which will inform you of the type of tool you're using for your analysis. So, and some works have been done on that, so we can easily replicate with those tools to achieve our aim.
Is it effective, like you're working with Barley here, and now you're working with insects here kind of thing? Does that have a big role in it?
Yeah, it's effective, yeah.
Okay.
I think we have time for one quick question.
I was just wondering if you could elaborate on the microbiome and how that kind of reversed or helped within the MS patient, and if you did any more research on that.
Sorry, I didn't get that.
You just brought up the point of the healthy diet and the microbiome with the MS patient.
Yeah.
And I was just wondering if you knew a little bit more about that or how that worked.
Okay, so basically, you know, our body is just like, our digestive system is just like a factory system. So the food we eat, it gives us the fuel. So the workers are the microbes. So whatever you eat will determine the type of energy that you get, or will determine the type of the effect it will have on your body. So now, similarly, so those foods, they have some certain microbes that actually help to digest those foods. So when you eat foods that are, for instance, if you eat meat, so there are different microbiomes that actually digest meat. So, and sometimes the issue of cholesterol and all of that, maybe the microbiomes does not really know how to break down those cholesterol, then it will kind of have some negative effects. So, but when you eat something like a plant-based food, perhaps you have some microorganism that helps to digest those bacteria well to provide energy. It's kind of similar. So it's similar with plants as well.
Okay, thank you very much. Next presenter today is Linden Haubrick. So Linden graduated from BU this past May with his BSc honors, where he majored in biology and minored in chemistry and psychology. His honors supervisor was Dr. Cushon, and his honors work was on Pests. And so Linden is going to take it away with his title.
So can waxworms be the problem, or can waxworms be the solution to our plastic problem? So in 2022, the plastic production rose to 400 million metric tons. And that number only continues to rise as the years go on. And 30% of that ends up in the watershed. Since the inception of plastics in the 1950s, humans have created 8.3 billion metric tons. So that's just a number to you. Let me put that into perspective. That's 1 billion elephants, the weight of 80 million blue whales, or 25,000 Empire State Buildings. So that's a lot of plastic we've created in 75 years. Polyethylene is one of the most common types of plastics. That's things like our plastic water bottles, our single-use plastics like food containers, grocery bags, washing detergent containers. And its characteristics are that it's durable, chemically resistant, and has a generally low cost to produce. So if you have a product and you want to package it, it's great for you, but it's terrible for the environment because it takes so long to break down. Sometimes it can take decades to even centuries to break down polyethylene. So that just leads to the accumulation of this in the environment. But researchers might have found our solution, and that's waxworms. So in recent years, researchers have found that waxworms are actually able to eat and digest plastics. So waxworms have a relatively simple life cycle, starting off with our egg, moving into the larval stage, down into the pupa, into the moth, and then starting that cycle all over again. And interestingly, the larval stage is the only stage they actually eat. So they have to gain enough nutrients and fat storage to enter into these next stages of life. And we've seen when they eat plastics, they're actually able to maintain that fat storage. So here I'm just showing you the different larval stages. So the different instar stages, L1 to L5. And to get to one larval stage to the next, they have to successfully molt. So they start off as an egg. And when they hatch, they're around 1 to 3 millimeters long. So to put that into perspective, a grain of rice is 6 millimeters long. So they're like the half of the size of a grain of rice. So they're really, really tiny and hard to see. Moving into, they can get all the way up to L5 and L6. I didn't have any L6 at the time for this picture, so that's why we're getting the L5. But they can get as big as 30 millimeters long, which is the diameter of a toonie. So they can get quite big and chunky. And in their larval stages, you actually can't tell the sexes apart. You can only tell them apart when they enter into the pupa and moth stage. And in their pupa stage, you have to have a microscope to tell them apart. So when we're looking at sex differences, we tell them apart in the moth stage. So how we tell them apart is their mouth structure. So if they have like a beak, a pointed, almost like a witch's nose, that's considered a female. And then if they have like a straight nose, like Voldemort, then we consider that a male. So their natural diet is honeycomb. They invade and damage honeybee colonies, causing millions of damage with honeybee colonies a year. And they're found all over the world. So wherever there's honeybee colonies, there's waxworms. So the black is where they're present, and gray is where they're absent. So why can they eat plastic if their normal diet is honeycomb? And it all comes down to the structure of the tube. So polyethylene is just repeating hydrocarbon chains, and low density polyethylene over here, they have long and short branches, prohibiting it from tightly compacting together, which is why it's low density polyethylene. And I'm showing you LDPE because that's the type of plastic waxworms eat. And then if we look at the structure of honeycomb, these also have hydrocarbon chains as well. So if you think about it, if I took off my glasses, they look relatively similar to me. So we believe that since they're able to digest this hydrocarbon chains, they have a unique microbiota and unique set of metabolic adaptations that allows it to digest LDPE as well. So proposed model of how they digest these plastics, it all starts with chewing and their salivary glands. So they have found that in their saliva, they contain oxidizing enzymes. That's the first contact of breakdown of the plastics. Then they have a symbiotic relationship with its intestinal microbes. So in past studies, they've done research where they've added antibiotics and then plastic-fed waxworms without antibiotics. And with antibiotics, they're able to digest less plastics, therefore kind of showing bacteria is needed to digest plastics in some sort of way. And we think this is because bacteria have plastic degrading enzymes like lactases and peroxidases. But in the environment, this takes a really long time to break down. And we see in waxworms that they break down plastics relatively quickly. So we think it's also a combination of their saliva, but also with their gut. Because we've seen in the past that there's certain enzymes that are in larger quantities in plastic-fed waxworms compared to honeycomb-fed waxworms, suggesting that these enzymes are really important for LDPE degradation in waxworms. So we think that's why they're maintaining their fat storage. And we predict when these enzymes are in lower quantities that they'll won't be able to maintain that fat storage anymore. So I want to get the whole picture. I want to understand what exactly the bacteria is doing, what enzymes are contributing, but also what enzymes the waxworm in its gut is contributing as well. So my first thesis question is, what is the microbial enzymatic machinery that allows for LDPE biodegradation in microorganisms? So what enzymes are the bacteria contributing to this breakdown? So my objectives are to identify these enzymes from waxworms, from the gut of waxworms, and then also do biochemical testing to see their ability to degrade plastics. So first, we'll collect guts from LDPE-fed waxworms. We'll do RNA and DNA sequencing. And then we'll take some of the, if we find any of the candidate enzymes that I was talking about earlier that we've known that can degrade plastics, we'll clone them into E. coli and do some biochemical testing from there. So right now, I'm just practicing gut dissections. So the red lines are showing where the foregut is, then the midgut, and this is where we think most of that digestion of plastic is happening, and then the hindgut. And this is where the formation of their frass or their poop begins to happen. So in previous works in the lab, we've had struggles getting enough microbial RNA. We've had too much host DNA host RNA when taking out the gut. So I'm doing practicing new techniques to see if we can get any more microbial RNA compared to host RNA. So the first technique I'm doing is called a flushing technique. So I'm cutting out the gut and then flushing RNA extraction buffer through it over and over again to try to get more of that bacterial RNA instead of having a lot of the gut in there, because I'll just throw the gut out after. And I predict it will take around probably 10 to 15 gut samples to make one sample. And then the second technique is the centrifuge technique. So I'm again cutting out that gut and then putting it into a centrifuge. And because the waxworm cells will pellet at a lower speed, I'll put it at a lower speed, and that's compared to the bacterial cells. So that should end up where the pellet contains the waxworm cells and the supernatant contains the bacterial cells. And I'll do that over and over again until I don't see any more waxworm pellet. And then whatever gets me more of the bacterial RNA or the microbial RNA, that's the technique I'll use going forward and complete the rest of the steps I was talking about previously. So my second thesis question is, what is the enzymatic machinery that allows LDPE biodegradation in waxworms? So I'll be doing gene knockdowns for that. And think of that as kind of your electrical outlet that you have too many things plugged into and you don't quite know what does what, but you want to unplug your lamp. So you just start unplugging things until you find the cord for your lamp. That's kind of similar to what I'm doing. So think of the different enzymes that I saw that were upregulated previously. I'm going to unplug one from the waxworm, and if it turns skinny, that enzyme had something to do with maintaining that fat storage. But if I unplug ADH and it stays fat, that enzyme probably didn't have that much to do with maintaining that fat storage as well. So my objectives are to start by knocking down CYP450, alcohol dehydrogenase, and aldehyde dehydrogenase. Those are part of the enzymes that we saw that were upregulated in plastic-fed waxworms. And these are really important for lipid oxidation. So these are the three enzymes I'm starting off with first. And then I'll do gene expression analysis. And then I'll look at the fat storage to see if it's changed or not. So my methods, I'll start off with siRNA injections of these 3 separate enzymes. So siRNA is developed specifically for my enzyme of target. It will be entered into the cell and come and form with the RISC complex. And then this gray passenger strand, the non-coding or the non-complementary strand will leaving you with the blue strand. And it will go off looking for its complementary mRNA. So think of that as complementary colors. Blue is complementary to orange. So it will go hunt down for the complementary color in orange in this case. And mRNA, think of that as the instructions for my enzyme. So they will complementary pair, and they will degrade and cleave the mRNA so that those instructions are no longer readable. So my enzyme will no longer be produced. From there, they'll either be fed in a plastic-fed diet or in a honeycomb-fed diet. And we'll be doing RNA extractions, sequencing, and differential expression. But we'll also be doing lipid extractions and lipid quantifications to look at the fat storage. So I'm currently practicing waxworm injections. This is what I like to call the waxworm sandwich. So it's where I literally just take a sponge, fold it over the waxworm, and use a binder clip to clip it in place, because these guys are really wiggly. So I need something to hold it in place so that way I can get the needle in. And right here, it's hard to see, but these are the prolegs, and that's where I'm injecting them into. And that's a zoomed up picture. So I'm aiming right for the center. So I'm using a 30 gauge needle, and I have to be very precise. And that, or else they'll die. There are other techniques, like force feeding them, but we don't want to do that, because if I force feed them and then accidentally grow their mouthpiece at all, that can prohibit them from eating plastics, which later on we won't get proper results. So this is the best practice for this experiment. So an overall overview of my thesis. So I'm looking at what exactly the microbial enzymes are using for plastic breakdown. So I'm doing the gut dissection, RNA and DNA sequencing, and then taking those candidate enzymes and doing that biochemical testing. But I'm also looking at what enzymes the waxworms are using for breakdown in the gut. So I'm doing siRNA injections, and then they're either fed a LDPE or honeycomb fed diet. And then we're doing gene expression analysis and fat storage analysis. So future directions. So these guys, you can't just like send them out into the wild to eat the plastics, because we have to remember these guys are pests. So they'll just go find the honeycomb and ruin honeycomb farmers' lives. But also, our plastic problem is too big to even do that. But if we understand what they're using to break down plastics, this can lead to new potential avenues for new technologies. So in summary, because LDPE and honeycomb have such similar structures, we believe that they have a unique set of microbiota and metabolic adaptations that allow it to degrade plastics. And my goal is to figure out what the microbiota and what the waxworms are contributing to this breakdown. So I want to thank Dr. Cushon, Dr. Loewen, and my lab mate Raya and NSERC for funding this.
We have quite a bit of time for questions actually. So hopefully there's plenty, and don't forget to grab some coffee also. Pretty.
How effective are your siRNAs for knockdown?
I haven't started those yet. I've just been practicing injections, because if I don't hit that, then they'll just like turn brown and die. So I'm practicing and making sure they survive before I even inject them.
You might pulmonary or something from other studies.
No, they actually like from the papers I've read, I've never seen them do siRNA injections, but they do the waxworms are a bacterial model, so they do it bacterial injections into them. So that's why we know that they can survive the injections. No one's just have done siRNA knockdowns yet.
Is your sponge technique enough to keep them from wiggling, or could you do it in a walk-in cooler so that they were just less active as well?
It's pretty good, but sometimes if they're really wiggly, I'll put them in the fridge for a minute and then just take them out and then do the injection. But usually the binder clip is enough to just like really just switch them in place and.
Hypothetically, if when you're doing these gene knockdowns, you find that none of any one particular gene of those three is contributing to the fat storage significantly enough that, knocking it down completely eliminates that. Would you try doing a knockdown of a combination of those genes?
We haven't talked about that yet because we're just kind of going, but yeah, I could see doing that as well.
Thank you.
Questions? They're eating LDPE, quote unquote. Do you think HDPE would be more nutritious for them?
I don't know about that. I just it would take them a lot longer to eat. I feel because LDPE is very it's like your grocery bag. It's very flimsy. It is durable, but it's more like the honeycomb. So it would probably take them a lot longer to eat it, but I'm not too sure about that.
Do they have to be encouraged by extreme hunger to resort to eating plastics or?
No, they'll just eat it. If they don't have honeycomb, then they'll just eat plastics. We have to keep them in glass jars because they'll eat right through the plastic, even though they have honeycomb in there. They're hungry guys.
Sorry, Aspen, seriously. So a lot of the issues with microplastics and the breakout of plastics is that they're going to be interfering with typical pathways or digestion or anything like that. Do you see this as being a potential issue in the future if you're having these things, these plastics, whether or not the actual particles or microplastics themselves will then actually impede the effects that you're actually seeing at a macro level, not at the level of the enzyme? They build up in the body and then they're inefficiently eating it or something like that, or can't even eat it, or damage.
Maybe. I'm not too sure though about that.
Or not.
Yeah.
Again, where you were getting into the bacterial genes, the RNA, and so your technique was designed to separate off and concentrate the bacterial RNA product. But there's some kind of signature on the product itself that says this is definitely bacterial and not insect.
And not what?
And not from the insect. Can you looking at the RNA itself or the DNA you get from it and say, because of this nature of the sequence, whatever, that you know it's definitely bacteria.
Yeah, we can tell the difference between.
How do you know?
They're different primers.
Okay, our next presenter today is Raya Turner. So Raya graduated from BU with her Bachelor of Science in 2024, majoring in the biomedical stream of biology and minoring in chemistry. She completed her honors thesis in the supervision of Dr. Kristen Loewen. Her thesis project examined the impacts of a sustainable insect diet on the survival, growth, and metabolism of larva to juvenile zebrafish. So today, she's continuing with zebrafish and is presenting her master's thesis proposal, which is to investigate the role of ERRa alpha in zebrafish metabolism, which I suspect is self-signaling. So get your way.
Okay, thank you very much, Mike. So these are zebrafish embryos. They are tiny but mighty tools used across the field of biology to learn about many different biological processes. Specifically, they can be used to study both development and metabolism. So what is our metabolism? It's occurring in every single one of our cells, typically breakdown or build-up processes. It can refer to many different areas, but I'm focusing on energy metabolism. So that is to do with the generation of cellular energy. So as you can see here, I've included a picture of a mitochondria, which I'm sure you've heard is the powerhouse of the cell. And that is true for the most part. So within our mitochondria, there are multiple different energy metabolic processes that are occurring in order to generate mass amounts of one form of cellular energy called ATP. So that happens in the presence of oxygen. There are also other metabolic processes that can contribute to our oxidative metabolism occurring within the mitochondria. One that you might have heard of is called glycolysis. So this is the breakdown of sugars or carbohydrates in our diet, specifically using the molecule glucose. It can be broken down into other products, sent into the mitochondria, and then further contribute to those energy metabolic processes under aerobic conditions. There are many other metabolic processes, and some are even the reverse of some of these processes. So the reverse of glycolysis is called gluconeogenesis, and essentially this is using energy to rebuild that glucose molecule depending on our environment, the situation, and when our cells might need it. So it's very situation dependent. So with the mass amounts of metabolic processes that we have occurring in all of our cells, it's essential that we have regulation and plasticity in order to achieve energy homeostasis. So energy homeostasis is essentially the balance of energy saving versus energy spending. Our energy homeostasis is maintained by constantly fine-tuning these metabolic pathways in response to environmental pressures and our situational needs. We call this metabolic plasticity. So you can think of metabolic plasticity a little bit like a volume dial. Depending on what's happening in our environment, we can turn up certain processes to suit our needs or turn down other metabolic processes when we don't need them to save that energy. So remember that our metabolism is very environment and situationally dependent. So we are responding to those changes, and in response, our metabolic processes are being fine-tuned in order to give us correct energy levels, normal responses, and an overall healthy cell and healthy organism. So that's when our volume dial is working correctly. When our metabolic plasticity and our regulation is not working correctly, it becomes difficult for our metabolic energy needs to be met. So, for example, this can lead to imbalanced energy, improper responses, and can actually lead to many different metabolic disorders. So if you think of type 2 diabetes, this is something that can occur within that disorder. We're not getting the energy that we require. I'm interested in one specific regulator of our metabolism. So you can think of this gene like the volume dial I was speaking of earlier. It's called estrogen-related receptor alpha, which for the rest of the presentation, I'll be referring to as ERRa. So even though its name has the word estrogen in it, it actually does not work with the estrogen molecule. That would be estrogen receptor. Instead, ERRa is referred to as an orphan nuclear receptor. So what this means is that to this day, we still don't know of any natural ligands or molecules that ERRa binds to in order to function. Instead, it works as a transcription factor. So a transcription factor is able to bind to certain parts of our DNA dependent on the sequence. So the ERRa protein finds that specific sequence, and with the help of other coactivators and proteins, it can actually act as a switch or a dial to turn up specific metabolic genes or turn them down depending on which factor you're considering. So effectively, we have control over our metabolic processes on the level of gene expression. So everything we know about ERRa has primarily been done in mammal models such as mice. And there are few studies in non-mammals or other animals such as fish. So we want to know whether other animals or other vertebrates are able to utilize ERRa in a similar way. Or in other words, does ERRa have a conserved function in the zebrafish? So you might be wondering, why am I using zebrafish? Well, they are a model organism, which means they are very popular, widely available, and they're used because they are a simple model in which we can learn about many different biological processes. So one of the advantages of using zebrafish is that they're transparent. So as you can see here, this is a zebrafish embryo at 24 hours post-fertilization. It has the developing eyes, developing body. It's surrounded by a chorion, which essentially acts like an eggshell to protect it for the first few days of its life. And it has a yolk. This yolk provides the essential nutrients it needs throughout the first few days of its life before it hatches. So going back to the transparency, this is an important aspect to consider if you're using zebrafish, because it allows the easy visualization of body development and organ development. So it makes it a lot easier to study these changes. They also have very high fecundity, which means that we can collect hundreds of embryos per week compared to certain mammal models where we're getting significantly less offspring from them. One last advantage would be their genetic similarity to other vertebrate species, including humans. So a lot of gene function studies are done on zebrafish, as they can tell us a lot of information about how certain human genes could function. So for my project, I want to explore the role of ERRa in zebrafish metabolisms for a few reasons. So the first is that from an animal physiology perspective, we really don't know that much about how other animals besides mammals are able to regulate their metabolism and respond to their environment effectively. So this is becoming more important as our environment is changing due to climate change and anthropogenic factors. It is of interest to understand how animals are able to be so resilient and survive the changing conditions. So I'll be exploring that in relation to energy homeostasis. On the other hand, zebrafish is a model organism. So as ERRa has been implicated in various disorders, human disorders, we would like to see whether the function of ERRa is conserved as compared to mammals. If we do find that the function of ERRa is similar in zebrafish, we could potentially apply this biomedical model in order to better understand the pathology of all of these disorders, such as type 2 diabetes and non-alcoholic fatty liver. On top of this, we can also explore treatment options for these different diseases. So for my project, I'm going to be studying the function of ERRa by reducing the activity of it. I will do this using something called a morpholino. A morpholino is a modified sequence of nucleotides, and the function of it is to block a specific protein from being made. So in my case, that will be ERRa. Typically, our genetic instructions are read by ribosomes, and the proteins are then built. When we introduce a morpholino into an organism, it targets the specific gene we've designed it to target and acts as a physical block from the reading of that genetic information. So the protein can no longer be decoded or read. So by reducing the amount of the protein or the activity of the gene, we can see what processes it controls. I like what Linden was saying with unplugging the different genes. So the first objective of my project is to successfully knock down or reduce the activity of ERRa in zebrafish embryos. The second objective is to observe the phenotype, so the physical characteristics or molecular signatures of the zebrafish embryos with reduced ERRa. My third objective is to determine the regulatory role of this specific gene in zebrafish metabolism. So exploring how it could be different than its description in mammals, or if it's similarly conserved. My first hypothesis is that zebrafish embryos injected with an ERRa morpholino will exhibit a reduction in both the ERRa transcripts or mRNA, as well as the activity. My second hypothesis is that embryos with reduced ERRa will exhibit differences in their phenotype through things such as their morphology, development, and then on the molecular side, they will have differences in their metabolism-related gene expression if it is a regulator of metabolism. My third hypothesis is that ERR alpha influences the expression of many different genes involved in mitochondrial metabolism. So it is a master regulator of zebrafish energy metabolism. So just a few predictions I wanted to go through before I get into my experimental design. I would predict that embryos injected with the ERR alpha morpholino exhibiting the reduced level of ERR alpha would have trouble growing, so they could have small structure or morphology or shape. So I can measure this with a microscope. I would also expect to see some delayed development in the group that has the reduced ERR alpha activity. If it is a master regulator, it should have problems with controlling these specific processes that we need to give us the energy that we require. Going along with the delayed development, I would expect that ERR alpha reduced zebrafish would have a decreased hatching rate, again for the same reasons. And then I would predict there'd be differences in the heart rate as the heart has high oxidative capacity. So if it is the same as mammals, it should be highly expressed there, causing issues in the decrease. On the molecular side, I predict that with reduced ERR alpha activity, I'll see a decrease in metabolic genes and activities. Going into some of my experimental design, it all begins with animal husbandry and care. So I start by breeding wild-type zebrafish. I place breeding traps, and then the next morning I collect the fertilized zebrafish embryos. I then move on to my microinjection stage. So I split the embryos into two groups, one to receive the control morpholino, which has no effect on the protein expression, and then the other group will receive our designed ERR alpha morpholino to reduce that activity. After that, I move on to visual confirmation and imaging. So each of these morpholinos is tagged with a fluorescent molecule, which allows me to visualize the uptake of the morpholino after injection. So I initially inject them into that yolk that I was talking about earlier that delivers all their nutrients. And by 24 hours, I should be able to see the fluorescent morpholino distributed to the rest of their body if it was successful. As well as that visual confirmation, I remove any embryos that don't exhibit this fluorescent, full body fluorescence. And I will also be using a specific zebrafish software in order to assess some of their structural differences. I'll then move on to my molecular confirmation. So just for a little bit of context, this is the lifespan of zebrafish for the first 5 days. I inject the embryos when they're in the 1 to 8 cell stage, and then I'll be sampling them at a few different time points, 24, 48, 72 hours post-fertilization. So once I've sampled these, I can use the samples in order to evaluate the knockdown itself. So to do this, I have to do RNA extractions to isolate that genetic material. I then change the RNA into a double-stranded molecule so that I can effectively work with it. I then use a technique called qPCR, which allows me to measure both the gene expression of the target that I'm trying to knock down. So I would expect to see a reduction in the ERR alpha gene expression for those injected with the ERR alpha morpholino. As well, I can use this gene expression analysis method in order to measure other targets that might be affected by the expression of ERR alpha. To further confirm the knockdown of ERR alpha, I'll be using something called a luciferase assay in order to measure the activity of ERR alpha. So that's just a secondary confirmation that the knockdown was in fact successful, and it wasn't just a reduction of the ERR alpha transcript. I'll also be using some different enzyme assays in order to look into the activity of specific energy and metabolism processes and see how they're affected by the knockdown of this specific gene. So the genes and activities of interest are mostly to do with our energy metabolism. So those oxidative energy metabolism occurring in the mitochondria. I've only included a few targets, but I do have a list of more. Some of these include citric synthase and cyclooxygenase 1, which are both involved in that mass production of ATP I was discussing. As well, I'll be looking at other breakdown processes that can contribute products to our mitochondrial production of energy. So one of these includes the breakdown of sugar or glycolysis that I was speaking of earlier, as well as the breakdown of fats that are introduced to our bodies from our diet. So this is referred to as beta oxidation. Moreover, I will be looking at a few different co-activators and transcription factors that have also been shown to contribute to these type of processes. So right now I'm at the beginning of my project. I've been injecting the embryos with either the control or the ERRa morpholino, as you can see here. They're all lined up. I do have to do quite a few microinjections, as it takes at least 20 successful embryos in order to get one sample. Unfortunately, I have been noticing some increased mortality in my group injected with the ERRa morpholino versus the control. So now I need to determine whether that concentration of the morpholino is accurate for my studies. So to start evaluating this, I was separating the morpholino injected embryos by fluorescence. So as you can see here, this one was injected with the control morpholino, and this embryo was injected with the ERRa morpholino. And essentially the idea behind this separation is that if they're exhibiting high fluorescence as well as a high mortality rate, my concentration of the morpholino is too high and I'm just getting too extreme of a reduction. So I do have some preliminary data here. So as you can see, I have the percent survival of those injected with the ERRa versus the control. And you'll note here that those exhibiting high fluorescence did have a much lower survival rate. So I then decided to begin microinjections with a lower concentration. So this is just some anticipated data just for like demonstration purposes, but I began with the 0.5 concentration. And I would expect that injecting them with a decreased concentration would still provide the knockdown of ERRa. So this is gene expression. However, it might be, their gene expression of ERRa might be a little bit higher, but essentially what I'm looking for is still that reduction of ERRa, but with increased survival of the group. So I began injecting them with the decreased concentration of the ERRa morpholino, and I will be continuing this for the next little while. I am noting, although I don't have data to show you today, that some of their physical development has been rescued with the slight decrease in concentration, as well as the survival has gotten a little bit better. So just to summarize for you, ERRa has been shown to regulate mammal metabolism, being coined a master regulator. However, we have few studies into its function in other animals besides mammals, or specifically rodents. We want to fill this gap in our knowledge in terms of how it functions in other animals. As well, we want to potentially be able to use the zebrafish and a model of deficient ERRa zebrafish to study this gene in different metabolic disorders in which it's been implicated in. So we could do this by using CRISPR technology. I'd like to thank Dr. Lemoyne for being an excellent supervisor, Sanel for her work setting up the microinjection station, Dr. Greenwood and Dr. Ardelli for letting me use their facilities, Linden for being a great lab partner, and our funders. Thank you.
Questions from the audience?
Yes.
How conserved is ERR alpha in other different mammals? And also, I know there's, I feel like it's in just alpha, or at least ERRs are.
Yeah.
ERR alpha.
It is highly conserved, so I would assume the function would be similar.
Like in insects.
Oh, in
Or
insects?
sorry, so in different mammals, they're fairly conserved.
Yeah.
And then how about other wildlife?
I have seen with Drosophila. Yeah, I haven't done too much research on other vertebrates, although I'm sure.
Maybe you answered this, you showed a picture with like, you know, some gene and then like the ERR alpha response element. Was that for like just the purposes of this talk, or is that like kind of a defined response element, in genomes? And if so,
I believe it's a defined response element.
and the genes kind of associated with those elements, are they mostly metabolic?
Yes.
Okay.
Yeah,
And did you say that, or did you say that?
I don't think it's a that.
That's right.
Got, Dr. Barry here.
I was just wondering, what are the natural like ligands for this receptor then?
There are no known natural ligands. It actually uses one of the co-activators that I mentioned, not very well, but PGC1 alpha. Yeah, going back to Dr. Lemoyne's favorite gene. It actually uses that essentially as a ligand when it's activating gene expression.
Or cholesterol.
I have not seen anything about cholesterol. Everything I'm seeing says no natural ligand, but that doesn't mean there isn't one.
Just a quick follow-up. Sorry, do you see when you're looking at hatching success or hatching rate? One's timed effect and one's.
To be honest, I'm not 100% there yet, so I could do either, really.
Like, would you pick a day and say this is the day that they're not hatched and these are gonna be effective, or we should let them go?
It would be, I think it would be hatching. I'm not sure. I'd have to look.
Success, right? 'Cause
Yeah,
sometimes
yeah.
if the rate
It wouldn't be a specific time point, it would be by a certain time, if that makes sense.
Okay, thanks.
One last question, has to be very quick.
Okay.
Are mutations in ERR alpha in mammals embryonic lethal? Like, is it possible it just is lethal? Or is it no?
No, it's not lethal.
It's not lethal.
I don't believe so, no.
And I believe that was going to be my question.
Okay.
Okay, thank you very much, Raya. Okay, our next presenter is Carter Williamson, who's a MELS student from Cypress River, Manitoba. Carter graduated with his BSc in biology from BU in spring of 2023. Carter's talk today is about metabolic adaptations and chemoresistance in triple-negative breast cancer. So looking forward to hearing about it.
Thank you, Dr. Schreck. Thank you, everyone, for being here. So just a little background. Obviously, as we all know, breast cancer, like every other cancer, is the abnormal excessive growth of cells. And in our case, breast cells. Cells will grow and proliferate and eventually travel throughout the body, continuing to grow, impeding organ function and ultimately causing fatality. It's estimated that 30,500 women in Canada will be diagnosed with breast cancer this year, making it the most commonly diagnosed cancer among Canadian women. It's also the second highest cause of cancer-related mortality among women. So current current research is focused on finding treatment options for later stage diagnoses. Obviously, we would prefer to find better early detection methods, but it's also crucial that we find treatments that are better at treating later stage diagnoses as well. So what are our current treatments? Currently, we have things like surgery, obviously, which involves the surgical excision of a tumor, and radiation, which is the local direction or directed application of radiation to kill cancer cells. But then we also have drug treatments which work systemically, such as chemotherapy and hormone therapy. The choice of treatment that we use largely depends on the stage of diagnosis and the subtype of cancer that one's been diagnosed with. Chemotherapy is more common in later stage diagnoses, and this makes sense, being a systemic treatment in later stage diagnoses, meaning by definition that cancer has spread systemically. But there are some major issues with chemotherapy drugs, as we all know and are familiar with likely, is the side effect profile for patients. And additionally, they also face issues with resistance, so they aren't totally as effective as we'd like them to be. So speaking of subtypes, we choose our treatment based on subtype. These are some examples of subtypes. The luminal A and B subtypes account for roughly 60% of all diagnosed cases, whereas HER2 positive or HER2 enriched account for roughly 10 to 15% of all diagnosed cases. And then triple negative breast cancer, which I'm going to be focusing on, accounts for 15 to 20% of diagnosed cases. And they're classified based on receptor status. So we see that the luminal subtypes are positive for hormone receptors, such as progesterone and estrogen, and they may or may not be enriched with HER2 receptors. HER2 receptor enriched lack these hormone receptors, but they are HER2 receptor enriched. And finally, triple negative breast cancer possesses neither. They don't have hormone receptors or HER2 receptors. And when we look at the prognosis based on case, we see it gets worse as we go down. And this is due to the fact that we lack targetable receptors. Receptors would be viable options for targeted therapies for triple negative breast cancer. So as I stated, later stage diagnoses, more aggressive cancers, typically we like to use chemotherapy. Triple negative breast cancer is aggressive, spreads quickly, so chemotherapy is very frequently used for triple negative breast cancer. And as I also mentioned, resistance is an issue, and they don't work as well as we would like. So 30% roughly of triple negative breast cancer cases reach a complete pathological response. So that's less than half of triple negative breast cancer cases, which are completely being resolved with use of chemotherapeutic agents. One such chemotherapy drug that we use typically for breast cancer is called doxorubicin, nicknamed the Red Death for its characteristic alarmingly red color. It's an anthracycline class of drug, so it's an antibiotic produced by bacteria, and it physically makes associations with DNA and RNA to prevent synthesis, but it also works as an inhibitor of topoisomerase 2, which is an enzyme required for DNA replication. So in a couple ways, it's preventing DNA synthesis, and in that way, it's kind of preferentially inhibiting DNA synthesis and rapidly dividing cells, which are more frequently dividing and are required to replicate their DNA. So chemoresistance in triple negative breast cancer, even in early stage triple negative breast cancer, we see that about 40% of cases develop resistance and result in recurrence. Once resistance has been developed, there's a very limited success rate for trying additional or alternative chemotherapeutic drugs at only 16 to 25%. So when we treat with doxorubicin in our lab, we found that there's an increase in this protein called COX-2, the expression of COX-2. So this is just looking at the expression change in several different cell lines. These are our cell lines. The M parental there is our lowly metastatic cell line far on the left, and we can see that this less aggressive cell line is showing not a significant change or increase in COX-2 expression, whereas the other cell lines, which are our highly metastatic cell lines are showing significant increases compared to the vehicle and 48 hours post-treatment with doxorubicin. And this has been also validated that was our gene expression, and that has also been validated via Western blot, the protein expression in several different cell lines as well. But what is COX-2? Why do we care? So COX-2 is an enzyme. Unlike COX-1, which we also express, COX-2 is expressed only during inflammation in tumorigenesis. COX-1 is constitutively expressed. COX-2 acts on arachidonic acid and results in the production of prostaglandin E2 or PGE2. And this PGE2 finally goes on to act on EP receptors. So this is like the signaling of inflammatory pathways. Our lab has looked primarily at the COX-2 EP4 pathway. So COX-2 and its product EP4, how it interacts with EP4, and it signals a couple of different things. So through the PKA pathway, it is preventing apoptosis. Through the PI3K AKT pathway, it is signaling for survival. And additionally, through the RAF-MEK pathway, we're signaling for migration and proliferation. So this inflammatory signaling is preventing programmed healthy cell death, apoptosis, and it's also signaling for survival, migration, and proliferation, which are all cancer phenotypes. And we see that when we inhibit this receptor, the EP4 receptor, we can significantly reduce the viability of triple-negative breast cancer. So following, this is just looking at cell viability. So percentage of surviving cells following different treatment conditions. When we treat with just doxorubicin alone, we don't see a significant reduction in cell viability. But when we treat with doxorubicin in combination with inhibitors of this pathway at different points, such as a COX-2 inhibitor or an EP4 inhibitor, we do see more significant reductions in cell viability. So we've established there's a direct link between treatment with doxorubicin and COX-2 EP4 activity, but we've also observed that COX-2 alters glucose metabolism in breast cancer. So cancer cell metabolism, cancer cells characteristically differ in their metabolism. So they, and this is to aid in the creation of energy and also the components required for the excessive cell division that they undergo. Normal cells in the presence of oxygen will utilize oxidative phosphorylation primarily for their energy source. Oops. Shown here. And then in the absence of oxygen, they'll use glycolysis. Cancer cells, on the other hand, even in the presence of oxygen, they heavily favor using glycolysis to create their energy. And this gives them the components for cell division and the energy for cell division. This is called aerobic glycolysis, or otherwise known as the Warburg effect, which is a characteristic hallmark of cancer. And this altered metabolism promotes oncogenic signaling, cell proliferation, and also metastasis. So we see in our COX-2 high cells that glucose metabolism, or in a couple ways that glucose metabolism is increased. So when we look at a couple of different cell lines, just grown in media, we see that, couple different cell lines grown in media. The MCX parental, the lowly metastatic cell line, it's not decreasing the concentration of glucose within its media as much as our highly metastatic cell lines, COX-2 high cell lines, which are more greatly consuming glucose from their environment. And we see this also validated with the energy production. So in these cell lines, they're also producing more ATP corresponding with the consumption of glucose. Additionally, we've shown that COX-2 expression alters the expression of different metabolic markers related to glucose. such as PDHA1, ATP5A1, and Tiger. And we've done that through RNA expression, comparing over here on the left, just showing the changes in expression, the pink being the COX-2 high cell lines, the black being the parental or not overly expressing COX-2 cell lines. And then this has also been confirmed in Western blots as well, showing that these proteins are significantly upregulated. So my research questions are, what happens to glucose metabolism during chemotherapy treatment? And if there's a significant change, is this shift in metabolism mediated by the COX-2/BP4 pathway? And if both of those go the way we'd like, we're going to see if we can resensitize resistant cell lines by blocking the action of COX-2. So my hypotheses, when treated with doxorubicin, metabolic markers related to glycolysis will be significantly altered in COX-2 high cell lines. And additionally, if we inhibit COX-2 or its downstream target EP4, we can resensitize resistant cells to chemotherapeutic agents. First off, we need to determine the metabolic response of doxorubicin cells, or doxorubicin treated triple negative breast cancer cells, and that involves acutely treating triple negative breast cancer cells that are naive to doxorubicin and measuring their metabolic response. Additionally, we need to determine the metabolic response of doxorubicin resistant triple negative breast cancer cells. So we currently have a line of triple negative cells growing in our lab that are chronically expressed to doxorubicin, and so they've built up a resistance to a relatively high concentration to doxorubicin, and we'll determine the metabolic response of them as well. And then once we've done this, we hope to treat with doxorubicin in combination with COX-2 and EP4 inhibitors and see how this additionally alters metabolism. First off, what's going to need to be done is to determine an IC50, the dosage for doxorubicin that I'll be using, and this is based on inhibiting proliferation. And Reed has previously in our lab, he's figured out the IC50 for a different cell line, which is going to be a baseline to work off of. But I'm going to be determining the IC50 for the triple negative cell lines that I will be using. This is kind of the different groups that I will have. So it will involve a control group here on the left, which is naive to doxorubicin treatment, and then collecting RNA and protein for analysis. Additionally, we'll have the doxorubicin group treated for 48 hours with doxorubicin, collecting RNA and protein from them. We'll also have a dox plus 3 days rest group. So this group will get treated with doxorubicin for 48 hours, give it 3 days to recover, and then having RNA and protein extracted from them as well. And then finally, collecting RNA and protein from our established doxorubicin resistant cell line. We also want to do some functional assays. So one of which is a migration assay. So I'll be growing cells with each condition in a well plate, creating a scratch through the cells which have grown to fill the bottom of the wells, scratching them with a pipette tip, creating a sort of wound, and then measuring the distance that they basically recover, close that wound within a certain amount of time as a measurement of the functionality or the migration of the cells. I'm going to also be using a COMET assay to measure DNA damage following doxorubicin treatment. So this involves collecting cells of each treatment, fixing them, poking holes in the membrane, and then using current to pull out damaged DNA. And we stain them, look at them under a microscope. And what we see is a bunch of these little spots that resemble a comet. So this over here on the right is the head of the comet, and that represents the cell body containing all of the nuclear DNA, undamaged DNA, whereas this tail is small fragmented damaged DNA that has been able to be pulled out of the cell through this process. And then we measure the amount of this DNA that has been pulled out, this damaged DNA, as a measure of DNA damage. And what do we expect to see? We expect to see alterations in glucose metabolism markers. We also expect that doxorubicin treatment is going to be less effective against COX-2 high triple negative cell lines. And we're hoping that inhibition of COX and or EP4 could enhance the effectiveness of doxorubicin. So discussion, we hope that this can help uncover some mechanisms contributing to doxorubicin resistance, especially related to metabolic alterations, and hopefully identify some different metabolic markers that could serve as targets for drug treatment in the future. It's also possible that we can find that the COX-2 EP4 expression may serve as a predictor for the outcomes of doxorubicin treatment for triple negative breast cancer. So the idea is that we can enhance effectiveness of doxorubicin and minimize toxicity, and this is through combination treatments. If we can find different targeted therapies to use in combination with chemotherapy that would make it work better, we can reduce the amount of chemotherapy drugs that we need to use, and thus reduce the side effects for the individuals, but also make the drugs work better for longer for more people. So this is significant because 30,500 people are diagnosed, but that means that so many more people are affected. Early detection of cancer is key, but in the case that we don't, improving treatment options for individuals with worse prognosis and later stage diagnoses is key. I just want to say thank you to the lab, everybody in the research methods class for their feedback, the funding agencies, but especially Reed and Dr. Montgomery, who've been a massive help thus far. Thank you.
Our local support first.
Do you have any, you said you're gonna like observe or measure like metabolic response. Is there any like assays that you have in mind right now? Or are you talking kind of Brady on that?
Like the functional assays for metabolic?
Yeah.
Well, like he did the, just like looking at the consumption of glucose, like the concentration within the media. I do have to talk with him more about that, about other options as well, but
Not sure if I missed or not, but did you actually mention the kinds of biomarkers for glucose metabolism you can look at?
yeah, LDHA, DHA1, ATP5A1, okay, like that. Yeah. Previously established ones by Brady that he had looked at. Okay.
But plenty of time.
Can you go back to a slide where it was talking about the glucose markers? I have two questions. So those are like the glucose markers?
These are the ones that Brady looked at. Yeah.
Okay, so like.
These are the ones that he ended up, like, no, this isn't a comprehensive list. But.
I guess I was just gonna ask, like, how many markers would you like expect to be upregulated if there is like the Warburg effect occurring? Like, would you see it like one, or how do you consider?
Well, since it's just like a general shift to glucose metabolism, like you expect like any like I'm not sure about an exact number of genes, but I'm sure it would be genes related to glucose metabolism.
Yeah,
Like,
I mean, maybe
there's
it's a
any
silly
one
question on my end, but like, I don't know. I was just curious.
Just to add up there, so really try blocking oxidative phosphorylation and see what are the changes, and then he did by glycolysis inhibition. And then saw if he uses both, what happens. So, and also oxygen consumption, so he has done, but out of, say, 25 different markers he studied, these ones stand out, so. Yeah, thank you.
No questions. Maybe I get to ask one. So with the COX-2, do you know, and it's okay to speculate here, is COX-2 playing a direct role in the glucose metabolism that you're seeing, or is it playing more of an indirect role?
Well, so like I showed the different signaling pathways that it acts on, the EP4 pathway. I would assume that it's probably a more indirect, like just because those are it's signaling of oncogenic phenotypes, and we know that when we're signaling to those oncogenic phenotypes that we see it shift. Like when we become more aggressive, we shift towards more glycolysis, that Warburg effect. So I don't think it's necessarily directly signal. That's my speculation.
Okay, last call for, we still have another minute here or two. Okay. No. Thank you very much, Carter. Okay, so last but not least, so this is where I have the pleasure of choosing my own student. So Hannah Zeigler-Blair graduated in 2023. The Bachelor of Science from BU, where she majored in biology, minored in chemistry. And then she started in the MELS program in January 2024. And today she'll be sharing her progress with us on investigating the hypermodified uridine in Bowen-Conradi syndrome.
Thank you, Mike, for the introduction, and thank you everyone for staying this long. So my project focuses on biochemistry. Biochemistry is the application of chemistry to the study of biological processes at the cellular level. It looks at the structure and function of biological molecules and forms the foundation for understanding all biological processes. One of the most important biological processes in biochemistry is the central dogma. This is where our DNA is transcribed into RNA via polymerases. This RNA is then translated into protein via ribosomes. Proteins are important for our growth, structure, and daily function. So you can think of the DNA as the master blueprint that is then transcribed into multiple copies, which are then handed out to the workers or the ribosome. So our research in our lab focuses on the ribosome. The ribosome consists of two subunits, a large subunit and a small subunit, which come together during translation to bind to mRNA. The ribosome then moves along the mRNA and reads its code and uses it to build proteins. The ribosome also has A, P, and E sites, which are involved in the process of translation. So we haven't always known exactly how the central dogma worked. In 1926, James B. Summer found that proteins beat up reactants in the body. In 1944, Oswald Avery and co. found that it's DNA that holds our genetic instructions. In 1974, George E. Palade showed that ribosomes are the ones that build proteins. And in 1989, Sidney Alton found that RNA also speeds up reactions in the body. And in 2005, diamond black fan anemia was coined as the first ribosomopathy. Ribosomopathies are disorders associated with reduced ribosome assembly. So we originally thought that any issues with the ribosome would be fatal, but we're now learning that there is a family of new diseases being linked to ribosome disorder, which are also associated with cancer. Because of this, ribosome research is speeding up in the biochemistry world, and it's increasing our understanding of the central dogma. Our lab focuses on Bowen-Conradi syndrome. Bowen-Conradi syndrome is a developmental disorder which occurs in the Hutterite population. It is characterized by reduced growth and results in death during infancy. If you look at this figure on the right, you can see there is a 20-month-old with Bowen-Conradi syndrome and a 4-month-old without Bowen-Conradi syndrome. So you can see that the 20-month-old and 4-month-old are similar in size, when the 20-month-old should be much larger. Bowen-Conradi syndrome is a ribosomopathy, which, as I mentioned before, is a disorder associated with reduced ribosome assembly and affects the translation of genetic code into proteins. It is due to a genetic variation in the DNA of an essential protein, EMG1, which causes an amino acid substitution. This ultimately affects the protein structure and therefore function. So just to give you an idea of where the issue in Bowen-Conradi syndrome occurs, EMG1 is a protein that's involved in building the ribosome. And the variation in Bowen-Conradi syndrome occurs at the DNA level, which is then carried through to the RNA level, and ultimately affects the structure and function of the EMG1 protein. So EMG1 is a methyltransferase, which means that it adds a methyl group. It helps build the small subunit of the ribosome, and it does this by chemically modifying a special site. This special site is a hypermodified uridine. The hypermodified uridine is located in the small subunit of the ribosome between the A and P sites. It helps form the structure of the ribosome and is important to ribosome function. So as the ribosome is being built, this hypermodified uridine undergoes a 3-step modification process. The first step is a pseudo-uridylation or change in orientation, where a uridine is rotated 180 degrees to form a pseudouridine. This is carried out by a SNO RNA called SNR35. The second step is a methylation or the addition of a methyl group carried out by the protein EMG1, like I mentioned before. The third and final step is an acetylation or addition of an acetyl group carried out by the protein TSR3. And the issue with Bowen-Conradi syndrome occurs at the second step here with EMG1. So just to kind of zoom out and give you the big picture, EMG1 is involved in the building of the ribosome by methylating that special site in the ribosome, which then allows the ribosome to go on and participate in the central dogma and build proteins. So my research question is, what is the role of the different components of the hypermodified uridine to the structure and translational ability of ribosomes? My project will consist of 2 parts, a structural analysis and a functional analysis. For my structural analysis, I will be carrying out antibiotic structure probing, and for my functional analysis, I will be carrying out dual luciferase reporter assays. I will be doing this in our model organism yeast. We use yeast in our lab because it is much easier to work with than human cells. There are experiments that we can do in yeast that we couldn't in human cells. There's also a strong conservation of eukaryotic biochemical pathways in yeast. And the Bowen-Conradi syndrome cells are difficult to obtain as there are very few children with the disorder. So jumping into my structural analysis, where I will be doing antibiotic probing, as I mentioned before. This is where I will be observing cell growth with different ribosome antibiotics in the ribosome binding antibiotics in the media. Altered growth of our cells then indicate that the ribosome structure has altered and suggest that antibiotics are binding to the ribosome differently and that the ribosome structure has changed. I'll be carrying this out structural changes can be exaggerated at different temperatures. So because of this, in addition to our normal incubation temperature at 30 degrees Celsius for yeast, I will also be using a cooler temperature at 17 degrees Celsius and a hotter temperature at 35 degrees Celsius. This is a list of my antibiotics that I'll be using. These first 5 target the small subunit of the ribosome, where the hypermodified uridine is located. And these 2 target the large subunit of the ribosome. I'll be looking at these antibiotics because the hypermodified uridine is located at the interface where the large subunit and the small subunit bind. I anticipate that G418 and paramomycin will be the most telling as they bind very close to the hypermodified uridine. So I will be doing my antibiotic structure probing using dot plates. This is basically where we have our culture of yeast on a petri dish. We then take this culture and culture it overnight in liquid. The next morning, we make sure that the cells are then of the same density and in the same phase before carrying out a serial dilution. And then we dot the serial dilution using a multi-channel pipette onto a petri dish. This allows us to see differences in growth. This is the plate map that I'll be using. So I have two controls. So my first line will be my first control there. I will then have my strain of wild type EMG1 cells. The third line will be my strain with the BCS EMG1 variant cells. I then have my second control. I will then have my strain where SNR35 has been deleted, which, as I mentioned before, is that first step. And my final line will be of my cells where TSR3 has been deleted, or that third and final step. So I hypothesize that antibiotics that bind closer to the hypermodified uridine will cause more of a disruption in growth to the altered strains. I predict that BCS EMG1 cells will have less growth than wild type EMG1 cells, and that SNR35 deleted cells will have less growth than the TSR3 deleted cells. So just jumping into my results so far. So my first plate that I have up here is my loading control, where we expect that everything will grow if we dotted the serial dilution accurately, which is what we're seeing here. I then have two plates with no antibiotic, one at 30 degrees Celsius and another at 35 degrees Celsius. So just kind of moving through the lines here. I have my first control here where we expect to see residual growth, which is what we're seeing. I then have my wild type EMG1 strain, which is exhibiting wild type growth. I have my BCS EMG1 cell strain, which is showing a slight reduction in growth compared to the wild type EMG1 cells. I have my second control, which is showing wild type growth, which is what we expect to see. I then have my strain where the SNR35 has been deleted, which is showing a slight reduction in growth similar to the BCS EMG1 strain. And then I have my deleted TSR3 cell strain, which is showing similar growth to wild type. So at this temperature, these are just kind of slight reductions in growth, but we can see that these have been exaggerated at the 35 degrees. So we can now see that the BCS EMG1 cell strain has a severe reduction in growth, and so does the deleted SNR35 strain. I then have two more plates, these now with antibiotics. So this first plate has the antibiotic paramomycin, which, like I mentioned before, targets the small subunit. And we can see that BCS EMG1 cell strain and the deleted SNR35 cell strain have little to no growth compared to the wild type EMG1 strain and the deleted TSR3 cell strain. I then have anisomycin, which targets the large subunit. And rather than targeted disruptions in growth, we're seeing that there's more of a broad disruption in growth. So this is where I'm at so far. My next steps will be moving through those antibiotics that I listed before, and then moving on to looking at the different temperatures. Now looking at my functional analysis. A past student in our lab showed that there is a difference in translational capacity between the BCS EMG1 cells and the wild type EMG1 cells. He did this using puromycin incorporation assays. So I will be delving into this difference and looking at translational speed and accuracy. I'll be doing this using dual luciferase reporter assays. So how this assay works is we give the ribosomes the code for two proteins, a firefly protein and vanilla protein, both of which produce light. And in between these two proteins, there is a challenge that the ribosome has to overcome. This could be a stop codon that the protein has to accurately recognize and stop translation, or a frame shift that it has to be able to overcome and continue translating. So the protein will move along, translate that firefly protein, which we can then measure the amount of light produced. And if it's able to overcome that challenge or recognize the challenge, the ribosome will then move along and will be able to measure the amount of light that the renilla protein has produced. So how this works is we again start out with our cell culture of yeast. We take the cells and wash them. We then lyse them or break them open so we can get access to the proteins. We then add a reagent which allows us to measure the amount of light corresponding to the firefly protein and renilla protein expression. We then add a second reagent which stops the first reaction and allows us to measure just the amount of light being produced by the renilla protein. So for this, I hypothesized that wild type cells will be able to accurately translate the mRNA with the challenge. And I predict that BCS BMG1 cells will have a harder time accurately translating the mRNA than wild type BMG1 cells. And I predict that SNR35 deleted cells and TSR3 deleted cells will have a harder time accurately translating different challenges. So just looking at some of my anticipated results, this is based on if there was a stop codon that the ribosome has to accurately recognize and stop translation at that first protein. So looking at the wild type BMG1 cells, I anticipate that we'll see a high amount of expression of the first protein, the firefly protein, and no expression from the second protein, the renilla protein. So the ribosome will be able to recognize that stop codon and stop translation. For the BCS EMG1 cells, I anticipate that the ribosome will first translate that firefly protein, but then it won't be able to recognize that stop codon accurately and will move along, and we will get expression from the renilla protein as well. For the deleted SNR35 and TSR3 cells, they're a bit harder to predict, so I don't have any anticipated results for those. So this is what I'll be doing after my antibiotic structure probing. So why is this research significant? Bowen-Conradi syndrome has a close to home impact as it affects the hydride population of the prairies. It's also a rare genetic disorder, which are not often researched. This research can also provide us insight into other areas. It'll help us understand the ribosome better. It also may have implications for cancer research, as a recent study found that hypermodified uridine has been found unmethylated in 50% of colon cancer patients. So just to summarize, ribosomopathy research is still in its infancy. Bowen-Conradi syndrome is caused by a genetic variation in the ribosome building protein EMG1. I will be investigating the hypermodified uridine in relation to ribosome structure and function. And Bowen-Conradi syndrome research could provide insight into other areas such as ribosome function and cancer research. I would like to thank my supervisor, Dr. Michael Charette, as well as my lab mates and our funders.
Any questions?
Could you go back to the dot plate slide briefly? Yeah, what I'm curious about is in some of the examples, like the no biotic at 35 Celsius, the Bowen-Conradi syndrome seems to have even less growth compared to the ones where SNR35 is deleted. Do you know why that might be the case, considering SNR35 seems to be a prerequisite for the EMG1 protein?
Yeah, that's because the EMG1 protein is essential and SNR35 has been found to be non-essential.
Yeah, and he also did some beautiful work on that over the summer. And the protein behaves beautifully, like you would expect, from the perspective that as the temperature goes up, proteins further destabilize and get less high protein. So at 35, it could get really, really sick. And at 30, it can hobble along better.
I might have missed that, might be also a stupid question, but it's because it's a human disease, human protein, we want to do that in human body temperature.
Not for a model organism. We do, Danny has also done work at 37 degrees, in yeast. But I'm using a yeast, version of the protein, and we also have a human version of the protein, which we then would use the 37 degrees.
Okay. What would you use that in?
What's that?
What would you put in that yeast? Don't try it.
Oh, sorry, we do also put it in yeast.
Okay, but it's fine at 37.
Yeah. It grows a little less, but yeah.
Sorry, with a hand go up somewhere.
I was just gonna. Like, so for the pheromycin, the control 1 doesn't grow, but then the control 2 grows, so what's the difference between your controls?
Yeah, so our control 1 is for the variance of the two proteins, and the control 2 is for the deleted, strains.
Oh, okay. So what is it exactly? Like,
The control 1
what's control 2?
it's the strain that we use for our deleted.
Oh, okay, sorry, I don't know what you used.
It would be kind of complicated to get.
I don't care that much. It's too late. It's not your.
Right,
I can tell you later.
We have to play games because EMG1 is essential,
Okay.
but SR35 is not. And so when it's
Okay.
essential, then like you can't just cut it out of the genome. You have to play games with it.
Okay.
Because
Hannah,
otherwise
I'm
it won't work.
interested. I just don't think I can handle the answer right now.
Hey, any last questions? And I know that everyone's tired, and probably hungry, and just so much that's.
Yeah, some of us are hungry.
Yeah, sure, they're sharing, it's awesome. Okay, there's no other questions, join me in thanking Hannah, but also everyone.